in cardiovascular and autonomic pharmacology. This award recognizes research excellence, scholarly impact, and scientific leadership in this critical domain. We are privileged to welcome the presenter whose exceptional research has brought them to this distinguished platform. Please, a big round of applause for them. We are delighted to have our chair, Dr. Ashok Dhatuselia. It is my privilege to introduce him. He is currently working as Associate Professor in the Department of Pharmacology and Toxicology at NIPER, Raebareli. He received his PhD from NIPER SAS Nagar and his postdoctoral training at National Brain Research Center, Gurgaon, University of Milan, Italy, and Department of Neuroscience and Pharmacology, University of Iowa, USA. His research interests include age-related neurodegenerative disorders, stress disorders, and neurobiology of metabolism. He significantly contributes to the work investigating neuroinflammation, diabetes-associated cognitive deficit, and brain injury. Dr. Ashok embarking in a new era of research focused on dissecting the trajectories for stress resilience and vulnerable behavior of an individual. He has published more than 90 publications in various journals of repute. Dr. Ashok has received several recognition awards from IBRO, ISN, Indian Pharmacological Society, ECNB, DST Serb, and ICMR. He has membership in many professional bodies, including the Indian Pharmacological Society. Welcome, sir. Now I would like to call our presenter. Our first presenter is Ashok Jangra from Central University of Haryana, Mahendragarh. Please come on the stage.
A very good morning to all. Myself, Dr. Ashok Jangra from Central University of Haryana. So I'm going to present my recent research work entitled Targeting ER Stress Alleviates DEHP-Induced Male Reproductive Toxicity, Behavioral, Biochemical and Morphological Evidences. So DEHP is the most widely used plasticizer, having a large production around 200 million per year, and around 2 mg of DEHP entered inside the human body per day. So there is a high risk of toxicity. So many research have been conducted to elucidate the role of DEHP in various organ toxicities. So we are focused on the male reproductive toxicity because 30% of the fertility is caused due to the male reproductive toxicity. So ER stress, we are focusing on the ER stress molecular mechanism. So our objectives were to examine the role of underlying the molecular mechanism of endoplasmic reticulum stress in male reproductive toxicity induced by DEHP. And then we validated the role of ER stress by using the specific ER stress inhibitor, that is 4-phenylbutyric acid. So this is our experimental plan. So we divided the animals, male vista rats, into 4 experimental groups, vehicle control, DEHP, and we have taken 2 doses of 4 PBA, 500 mg per kg and 1000 mg per kg. And after 28 days of exposure, we conducted the sexual behavior and copulatory behavior, and afterwards we sacrificed the animals and performed the biochemical Western blotting and the morphological by SAM and the histopathology. So in the results, we found that the DEHP significantly reduced the testis waste, epididymis weight, and gonatosomatic index, which was significantly prevented by the 4 PBA. Then we performed the sexual behavior, and as you can see that the DEHP reduced the time spent in the female zone, which was significantly prevented by the 4-PBA. Then we performed the copulatory behavior. The parameters we taken like latency to first intermission, mounting latency, ejaculation latency, intermission frequency, and mounting frequency. So all the parameters of DEHP treated rats, they are significantly altered, which was prevented by the 4-PBA treatment, both the doses 500 as well as 1000 mg per kg. Then we performed the testosterone estimation in the serum and the oxidative stress parameters in the testes, and we found there is marked reduction in the testosterone as well as there is increased oxidative insult by the DAHP exposure. So you can see that 4-PBA might significantly reduce the oxidative stress as well as they augment the testosterone level. Then we counted the sperm in different groups, and here you can see that DEHP 500 mg per kg significantly reduced the sperm count as well as the number of abnormal sperms was significantly increased in case of DEHP, and you can see that 4-PBA significantly reduced the number of abnormal sperms. So afterwards we performed the SAM analysis of the sperm sample, and you can see that in the DEHP there is bulging in the sperm, and we also found that there is presence of acephalic sperm in the DEHP, and there is coiling of the tail of the sperm. Then this is the picture of 4-PBA, and you can see there is normal sperm in the DEHP. So this is the SAM analysis of the testes, and you can see that in the DEHP, the red arrow show the blunt and wrinkled surface of the seminiferous tubules, and you can see there is significantly improvement in the 4-PBA treated group. Then we performed the histopathology of epididymis as well as the testes, and you can see that there is loss of interstitial as well as the lumen. There is loss of luminal diameter, so there is empty lumen, so it indicates that severe toxicity induced by the DHP exposure. Then the structural damage we also evaluated with the help of image J, and you can see that seminiferous tubule diameter, area, epithelial height, both all are significantly reduced by the DEHP exposure. Then the width of tunica albuginea, which is a protective layer in the testes, was also decreased, and the number of Leydig cells were also decreased, which is responsible for the low testosterone in the DHP treated group. So the number of sperms as well as there is increased sperm DNA fragmentation in the DHP treated group. Then we performed the ER stress related protein system blotting, and you can see there is increased upregulation of the GRP78 caspase as well as the CHOP in the DHP treated group, which was significantly prevented by the ER stress inhibitor 4-PBA. So in the conclusion, there is marked alteration in the spermatogenesis process as well as DHP induced the marked testicular damage, which was significantly prevented by the 4-PBA through inhibition of the endoplasmic reticulum stress. Thank you.
Open for the discussion now. Any good questions? Okay, that's right. It is Ashok Jain, right?
Yes.
Very good presentation.
Thank you, sir.
And you said it is mainly DEHP.
Yes, sir.
Now, you have measured the testosterone level
Yes, sir. In the serum.
and you run a normal or standard one.
Yes, sir.
And compare, and what is your final way of thinking when you brought to the conclusion?
Yes, sir.
What do you want to outcome of this study?
Outcome of the study is there is a role of ER stress in the DHP-induced male reproductive toxicity, and when we targeted the ER stress by specific ER stress inhibitor, there is marked improvement in the testicular function as well as the sexual behavior function.
Okay. Simultaneously, did you also look towards seminal recipe? Any in vitro studies?
No, we didn't perform.
That is also in vitro. You can run simultaneous and see to it because it has been done in which animal?
Animal male Vista rats.
Okay. So possibly
Definitely, sir. We will take into the.
So what are the primary organ when DEHP is deposited? Is the testis are the primary organ?
There are several studies which indicate severe toxicity in the liver, in the heart, in the brain, in the testes. But we have not measured the DEHP level in the testes. But we are planning to estimate the DEHP in the testes in the future studies.
Thank you.
Thank you.
From Naipa Library.
See, you should always respect the spirit of science. All the things are second. Spirit of science should prevail. That is what is the inner core of any research. Spirit of science should be. Go ahead, please. It is easy.
So good morning everyone. Myself Naval Kishore Joshi, and today I am will be our project work, like we can say our research work entitled as the 1-methyl D-tryptophan ameliorate the fair memory defect in the food stress model in rats. Next. So starting with the introduction, like what is we understand with the PTSD. So like PTSD is the condition which is triggered by the trauma induced by the several reasons. Either there is the war condition, or we can say it is the sexual trauma, or anything is trauma which triggered the PTSD, and it is characterized by the hyperarousal condition, avoidance, as well as the anhedonia conditions. So there are the several reasons which are involved in the PTSD development. One is reasons like medial prefrontal cortex as well as the hippocampus. Like medial prefrontal cortex is responsible for the emotional regulation, as which alter in the condition of the trauma, as well as the hippocampus, which is the consolidate the memory for these events. One of the major pathway involved in these conditions, like the which we can say the IDO pathway, which is responsible for the increase of several cytokines, as well as the development of TLR, development of the PTSD condition. Here we focus on the our drug, like one is the 1-methyl D-tryptophan, which is the selective IDO inhibitor, which we use for the inhibition of this IDO pathway for the cope up of the depression or with the condition of the PTSD. So here we hypothesize that when we give the stress, or the individual we suffer with the stress, there is the activations of the macrophages which is responsible for the release of TNF alpha, or as well as the IL-6, the inflammatory cytokines, which are follows the IDO pathway for the metabolize of the tryptophan into the following kynurenins, as well as tryptophan is the mainly precursor for the synthesis of the serotonin, which is we can say the hormone for the reward processing. So here we give the 1 MT at the different doses to check the effect of 1 MD to inhibit the IDO, and the effect of 1 MT into the PTSD conditions. So aim of our study was to evaluate the effect of 1-methyl tryptophan on the PTSD like behavior defect. To study to completion of our aim, to achieve our aim, our objective was to study the effect of 1-MT on the behavioral outcomes, oxidative stress, neuroinflammation, and the NMDA receptor disruption in the stress model of rats. So here we designed that, like after the 7 days of acclimatization, we introduced the animals for the stress development of stress. We can say the induction of the stress through the food shock stress model, and after the 24 hour of stress induction, we perform the neurobehavior at the 24 hour as well as the 7 day to confirmation of the result, as well as we give the co-treatment after the FSS exposure with the 1-MT at the 3 different dose, 10 mg per kg, 30 mg per kg, and the 100 mg per kg of body weight. So our next coming to the results, our our result indicate that the effect of FSS into the anxiety-like behavior, where we see that there is the increase into the anxiety-like behavior into the FSS, we can say the FSS exposure group, which is significantly reduced or reversed by the treatment of 1 MD as the dose dependent manner, as well as we can see that at the 100 mg of body weight, the dose at the 100 mg of body weight. Next, we check the different IDO enzyme activity as well as the cortisol level for the confirmation of model generation, where we can see that there is the increase into the, we can say the cortisol level into the serum, which is significantly reversed by the treatment of 1 MT at the dose of 100 mg per kg. As well as we also check the enzyme activity at the 24-hour or food shock stress exposure as the 7. We also check at the 7 day of food shock stress exposure, where we see that there is increase in the IDO enzyme, which is responsible for the development of PTSD in the, or we can say the memory consolidation into the hippocampus region mainly, which is significantly reduced at the 6 day as well as the 24-hour day by the treatment of 1 MT at the 100 mg of body weight dose. Next, we perform the 1 MT expression, or we can say we perform the pathway of the check the involvement of IDO through the NF-kappa B and the TNF alpha involvement into the development of PTSD, where we can choose the two different brain regions. One is the medial prefrontal cortex and the hippocampus. The reason of choosing these regions, like the medial prefrontal cortex is the responsible for the emotional regulation, or which is mainly involved into the PTSD condition, as well as choosing the hippocampus region is because in the PTSD condition the pair memory is generated, or we can say spatial memory is disrupted. So the hippocampus region is mainly involvement into the memory consolidation. So we choose the two brain regions where we check the this IDO pathway, where we can see that there is significantly increase into the FSS group, which is significantly decreased, we can say reversed by the 1 MT at the 100 mg of body weight. Further, we confirm the result with the immunohistochemistry. Next, we check the correlation pathway between the TNF alpha, IDO, as well as the NMDA modulator. NMDA, basically we can say the NMDA receptor. NMDA glutamate receptor are the responsible for the, we can say, stress vulnerability, which prevent to the animals from the conversion of the individuals from the vulnerable to resilience, to involve the to show the cope-up mechanism. So here we check the correlation between these mechanisms, like in TNF alpha and NF-kappa B, IDO, and the glutamate receptor. So here I conclude that the 1 MT treatment at the dose of 100 mg per kg shows the good response to cope up from the stress vulnerability and move toward the resilience. Thank you. Josie. Yes.
Okay, good work, Murad. You said IST also in between. Okay?
One.
IST.
Yes, sir.
And what could be the receptor subtype in that?
5ST.
Because it's the n number of receptors over there, at least up to 7. What could be the one specific one?
No, sir. We are not checking the 5ST in
Okay. You are only into NMD.
NMDA receptor.
NMD is also associated with what? Another learning and memory.
Yes, sir.
That's one more thing I just want. You said inflammatory biomarker. So what are the D1 which you would say, which can lead to this stress level?
Sir, cortisol.
Beyond that, you made 3, 4,
Yes, sir. MDA, we can say that the oxidative stress we also check for.
Sir, anybody wants to, sir?
Sir, glutamate receptors are mainly responsible for the stress vulnerability. The hyperactivation of the NMDA receptors are responsible for the development of stress vulnerability, which prevent the animals, or we can say that which prevent the individuals to convert the resilient to vulnerable groups. That's we check the role of glutamate. Hyperactivation of these receptors are preventing individuals to convert it into the resilience or not. So we can see that the increase of the glutamate receptor are preventing them to, we can say the individuals to convert into the resilience group, which are successfully cope up to the stress. Yes. For that, sir, we check the oxidative stress markers, where we can see that there is in stress condition, the oxidative stress markers and the reactive oxygen as well as the cortisol is higher in the stress groups. So this is significantly reversed by the 1MT treatment with the 1MT. Yes, sir. Sir, in the study with the 3 doses, we didn't see any toxicity till now. But the rationale of the choosing 3 different doses, like we go with the literature, so where we can see that there is the good results, we can say depict at the dose of 100 mg per kg. So we choosing that drug for the inhibition of IDO here.
Yes.
No, sir. Thank you, sir.
That's right. Sir, that is because we don't want to say the number. Yeah. So let us be uniform to everyone. I think it's one person.
So that we can manage the time.
Go ahead. Don't worry. Sometimes, sometimes depending on the nature of the work, you have to come out with some scientific explanation. That's good. Okay, please go ahead.
Now I honor to introduce the esteemed wife of Shri Manjeet Singh Ji, Shrimati Raminder Kaur, who join us today as a symbol of his enduring legacy and commitment to excellence. We are grateful for her presence as we commemorate this prestigious award, Professor Manjeet Singh Award. Let's a big hand of applause for her. Thank you, ma'am, for coming. Thank you so much.
Yes, can I add one word?
Please, please, sir. Please, sure, sir.
Student of the
Sir?
late Subhash Verma,
Sir.
Subhash Chandra Verma.
Sir.
And Subhash Chandra Verma doesn't matter. Dr. S.C. Verma and Manjeet Singh sir used to be, I think you worked along with Subhash Verma also. My guide R.K. Goyal, myself, we did work with Subhash Verma and Manjeet Singh sir, I think if I remember, more than rat uterus. He has worked on that and yeah, that's it.
मैं बस 2 चीज़ बोलूँगा, ज़्यादा नहीं बोलूँगा। देखो भाई, तुम नए नए बच्चे, मैं मंजीत सिंह सर का स्टूडेंट हूँ और वो हमारे भगवान हैं, हमारे गॉड हैं। वो कुछ नहीं बोलूँगा। उनकी खासियत क्या थी मैं कुछ कह ही नहीं सकता। मैं दिल से बोल रहा हूँ। आई एम प्रोफेसर इन एम्स। आई एम हेडिंग मोस्ट ऑफ द यू सी द मीटिंग फिटिंग जो वे चेयर फेयर, बट आई नेवर सीन पर्सनालिटी लाइक द मंजीत सिंह। क्या नॉलेज थी उनको, क्या डेप्थ में वो पढ़ाते थे। और सुनो, एट दैट टाइम मोस्ट ऑफ द फार्माकोलॉजिकल रिसर्च वाज बेस्ड ऑन द जस्ट फार्मास्यूटिकल एक्सट्रैक्ट एंड इट वाज, 2 मिनट रुक जाओ, इट वाज वेरी चैलेंजिंग to start molecular pharmacology at the standard international level in India, and he did this job. Where? In a small city, Patiala. And at that time, the lab of the Patiala was recognized as the Manjeet Singh lab. So, Indian Pharmacological Society started this prize for the best work in the molecular pharmacology in the name of the Manjeet Singh. Thank you very much, beta. Thank you. Thank you, sir. Thank you, sir.
Please go ahead, Srikanth ji.
Good morning, everyone. First of all, I would like to thank the organizers for giving me the opportunity to talk about our work on role of RIPK3 MLKL signaling in the development and progression of kidney fibrosis during CKD. So CKD, or chronic kidney disease, is not one disease. It is rather an umbrella term used to cover CKD from multiple etiologies. Importantly, fibrosis remains the common pathomechanisms in all these CKD. So what happens in fibrosis is the scar tissue develops, the kidney shrinks over the period of time, resulting in progressive decline in renal function and end-stage kidney disease. We have been working on necroinflammation, which is an auto-amplification loop of regulated necrosis and inflammation. So these processes activate each other, and this is one of the main through several studies we showed that this is main mechanism for AKI development. Particularly the necroptosis pathway, RIP3 MLKL signaling is very important. So in this study we wanted to know what is the role of persistent necroinflammation in development of CKD. So what we first did, we developed a model of CKD by feeding high oxalate diet to the mice, and we checked the expression of the RIP3 kinase, RIP3 MLKL. We found that the expression was increased along with increased alpha SMA and collagen 1, meaning there was also fibrosis in the kidneys. So based on that data, we hypothesized that RIPK3 MLKL may regulate the TGF beta signaling and regulate fibrosis in the kidneys. So to test that hypothesis directly, what we did, we developed CRISPR lentivirus to suppress RIPK3 and MLKL genes in the kidney. So we injected the virus intra-renally direct in the parenchyma, and then we developed the CKD model in those animals. So as expected, RIPK3 MLKL was suppressed in the kidney, and there was also decrease in the alpha SMA collagen 1, meaning there was less fibrosis. We also confirmed those results by immunohistochemistry for alpha SMA, which also showed less fibrosis. There was also reduced tubular injury in the kidney, and this all was associated with improved renal function in the animals. So this was of course the genetic proof, so we wanted to test pharmacological inhibition as well. We used inhibitors of RIP kinase 3, RIP kinase 1 pharmacologically, and we had similar results in that. So I'm not showing the same data again. So next we wanted to understand what is the molecular mechanism. So we perform bulk RNA sequencing in those kidneys, and interestingly we found that all the mitochondria associated gene signature was suppressed using gene set enrichment analysis. You can also see the heat map for the GO term mitochondria. So all the mitochondria genes were suppressed in the CKD. So this suggested that mitochondrial dysfunction happens. What we did next, we isolated mitochondria from the kidneys and we checked the expression of RIP kinase 3 and MLKL, and of course this expression was increased in the mitochondria. So RIPK3 has two known targets, MLKL and CAMK2. CAMK2 has been known to regulate the TGF beta signaling. So we also checked the activation of CAMK2 in mitochondria, and surprisingly we found that CAMK2 activation happens within the mitochondria in the kidney. So now we wanted to test pharmacologically what is it really regulating fibrosis in the kidney. So we did pharmacological inhibition of CAMK2. We used KN93, which is a chemical inhibitor of CAMK2. We treated these mice with KN93, and we checked the fibrosis markers, alpha SMA collagen 1 as well as TGF beta signaling markers, specifically the activation of phosphorylated SMAD2, SMAD3. So when we suppressed CAMK2 with KN93, we saw there was less CAMK2 activation, less TGF beta signaling, and less fibrosis in the kidneys. We also confirmed the result with immunohistochemistry for alpha SMA, which showed less fibrosis. There was also less tubular injury, and again this was associated with improved renal function in the animals. So based on this data, we conclude that the chronic or persistent necroinflammation drives mitochondrial dysfunction and kidney fibrosis during CKD. And through these studies, we identified RIPK1, RIPK3, MLKL, CAMK2 as novel biomarkers or novel targets to suppress kidney fibrosis in CKD. finally, I would like to thanks my PhD student Anjali, who really drove this work, and our collaborators. This work was performed in CSIR CDRI, so I also acknowledge the funding from CSIR CDRI, from DBT, and from BITS Pilani. No, that's it. Thank you.
How does chemical modeling kinase 2 activate downstream signals to affect CGF-beta? What is that particular part?
It's a very interesting question, sir. So what we believe that CAMK2 activation causes the or is involved in mitochondrial dysfunction and ROS production. ROS production phosphorylates the TGF-beta signaling. Now, whether that exactly happens within the mitochondria or outside, because SMAD is a cytosolic protein, so that's the missing link which we are following now in the following project.
So when you do CKD with oxalate, you think because there's more oxalate crystal in the kidney and kidney becomes sick and sick?
Very good question, sir. Again, because of the time limit, I couldn't show all the data, but we perform an independent model, unilateral ureteral obstruction model, which is another standard model for CKD. And we did all these experiments in that model as well, just to confirm whether this is model specific or not. And beyond that, we also checked the publicly available human kidney fibrosis data sets, and we find the increased signature of RIP3, MLKL, the necroptosis signaling in human disease as well. So this is, I believe that this is very translatable. Thank you, sir.
In fact, to suggest you, Srikant, as Saroj said, in fact today, CKD, we can even retarget some anti-convergence. That could be a combination interactive state.
Very good morning, everyone. I'm Harapriya Baral from the Department of Pharmacology and Toxicology in IPSCON Library. Thank you, everyone, for giving me this chance to present our research work, which is entitled as TRPC5 inhibition ameliorates the oxidative stress, neuroinflammation, and apoptosis in traumatic brain injury. So, traumatic brain injury is a sudden injury to the brain because of any physical external force, which can lead to temporary damage to permanent dysfunction, and it is one of the leading cause of death and disability worldwide, which affects more than 50 millions of individuals each year. And the causes are very common, like we face in the day-to-day life, like road accident and falls, etc. So pathophysiology of TBI includes primary injury and secondary injury. So primary injury is the initial events that happens just after the impact, which includes tissue damage, axonal shearing, triple B damage, etc., which further mediate the secondary cascade, which includes neuroinflammation, excitotoxicity, brain damage, etc. So the TRPC5 channel gives a TRPC5 channel is one of the modulator of the secondary injury. So our focus was to study this effect of some TRPC5 channel blockers, particularly clamizole. So whenever there is a impact or brain injury, the TRPC channel is activated, which increase the influx of the calcium. This excess calcium will increase further mediates the further mechanism like oxidative stress, glial cell activation, immune cell infiltration, and upregulation of various inflammatory markers, which further amplifies the neuroinflammation and apoptosis. So we analyzed the in silico study to see the clemizole targets TBI related pathway or not. So you can see the TBI and clemizole, they shares more than 160 common gene, and we made protein interaction network from this, and further we analyzed the top 10 hub genes, which includes TNF alpha, BCL2, caspase-3 and MMP9, which all are related to the neuroinflammation and cell death. Further, the cluster analysis revealed that TRPC5 is one of the major target from this network. Further, the pathway enrichment analysis revealed that cellular response to cytokines and programmed cell death is involved in the biological process, and PI3-AKT signaling is one of the important pathway involved in this particular gene targets. So further, for validation, we performed the in vivo study. We induced the animal for TBI with the weight drop model, and the study design is for 7 days. We given pre-treatment before the induction of the disease and continue till the 7 days. Meanwhile, we have performed various neurobehavioral assessment, and after 7 days we sacrifice the animal for the further biochemical and molecular estimation. So this neurological severity score, we can see the day 1, day 3 and day 7, significant increase in this score in the TBI group as compared to the control, which is attenuated by the clemizole treatment in dose dependent manner, that is 10 mg per kg and 30 mg per kg. Similarly, the locomotor activity also reduced, which is in case of TBI, and which is restored by the clemizole administration. Furthermore, we assess the spatial learning and memory, which shows increased escape latency and reduce in the time spent in the target quadrant in case of TBI, which is restored by the clemizole treatment. Similarly, we have seen the anxiety-like behavior in TBI, which is significantly reduced in the time spent in the open arm, distance traveled and number of entries in the open arm, which suggests that TBI group shows anxiety-like behavior, which is further reduced by the clemizole treatment. So collectively this data represent the clemizole amyloidous neurobehavioral impairment in TBI. Furthermore, we check the effect of the clemizole in neuronal death and triple B damage. So histological study, we can see the neuronal density in the cortex is significantly reduced in case of TBI. However, the treatment restored the neuronal density. Similarly, the triple B junction markers also, like ZO1, occludin and claudin 5, they are significantly reduced in case of TBI. However, clemizole treatment dose dependently restores this level. And one more marker like MMP9, which is a proteolytic marker, which seen to be increased in case of TBI. However, clemizole treatment reduced its activity. So collectively, this data represent clemizole reduced the cellular damage and preserved the triple B integrity. So furthermore, we check the neuroinflammatory markers in TBI. Various neuroinflammatory markers like COX, COX2, INOS, TNF alpha, IL-1 beta, IL-6 were elevated. However, clemizole treatment reduced these inflammatory mediators. Similarly, the oxidant levels also like MDA and nitrate levels also increased in TBI. However, clemizole treatment reduced these levels. We also check the apoptosis effect of clemizole in apoptotic pathway. So this apoptotic markers like BAX, caspase-9, caspase-3 are elevated in case of TBI. However, clemizole treatment reduced this level. So one of the pathway is PI3-AKT, which is known for the anti-apoptotic pathway. So PI3K and AKT phosphorylation is reduced in case of TBI. However, clemizole treatment restored this levels. Similarly, in tunnel histological study, we can see the number of dead cells are restored by the clemizole treatment. So our main target was to see the TRPC5 expression. So TRPC5 expression and co-localization are also significantly increased in case of TBI. However, clemizole reduces the expression. So from this we can conclude that clemizole shows the neuroprotection in various by controlling various mechanisms, and it emerged as a new therapeutic intervention for the TBI. Thank you. Yes.
And that is not the clinical situation.
Yes. Yes.
The time patients come
Yes, yes, sir.
So, have you examined the effect after PPI, the effect of this antagonist?
Sir, actually it is like not clinically not relevant that we can't give pre-treatment. It is just to evaluate the neuroprotective effect of the clemizole because its half-life is less. So we wanted to check whether it has any effect like immediate effect because primary injury and secondary injury, it starts just after the injury. So just to analyze its effect, the early event we give the pre-treatment.
Excellent presentation. Yesterday, I heard seen the same presentation by Dr. Sham Sundar Sarma. Are you part of that team? Exactly the same presentation.
Please, sir, that is in Parkinson disease.
Exactly.
Yes.
So I was about to ask. Many of the slides were same, but that is also from NIPER. That is also from NIPER. So, okay. So, apart from clemizole, what are the other agents that are affecting the pathway? Have you evaluated any other agents?
No, sir. Currently, we are not.
There are some alternates. So, see, I ask all the participants, when you present here, please look at clinical implications finally. She is the only one who has presented clinical implication. Henceforth, everybody, please try to correlate to, it is not just, pharmacology has to relate to disease. That is extremely important. Okay, good work, by the way. Thank you.
Thank you so much, sir.
MMP 9 on
A very good morning to one and all present over here. I am Pooja Singh. Today I am here to present my work on pormorphin, a smoothin receptor agonist, attenuates dexamethasone-induced suppression of neurite outgrowth in neuro 2A cells. Hypercortisolemia is a condition characterized by prolonged exposure to the elevated level of cortisol, arises due to dysregulation of HPA axis, majorly involved in the body response to the stress. Under persistent stress condition or prolonged exposure to the glucocorticoid, such as dexamethasone, can disrupt the mechanistic function and also increase the level of cortisol, which negatively affect the neuronal function and structure primarily because of neuronal outgrowth. Sonic hedgehog signaling is a signaling which is responsible for neuronal differentiation, neurite outgrowth, neuronal architecture, and the major core component of this signaling is patch 1, GLI and SMO. In the activated state, the SMO activates and leads to the translocation of GLI to the nucleus where it performs different functions and activate the BDNF, SOX2 and profilin, which are majorly responsible for neuronal architecture. From this, we can say that there was no significant study was there which are linking the mechanistic link between how the sonic hedgehog signaling and is altered during the prolonged cortisol condition, means under the hypercortisolemic condition. To address this question, we are hypothesizing that is dexamethasone, a glucocorticoid, which synthetic glucocorticoid may regulate the sonic hedgehog signaling along with the AKT-GSK pathway and alter the neurite outgrowth. For the induction of stress-induced model, we have used neuro 2A cells and exposed our cells with 1 micromolar dexamethasone for 24 hours. In the treatment strategy, we have used pormorphin, a smoothin receptor agonist, to overcome this effect or not, and we have incubated our cells for 24 hours and performed different examination like Western blotting, ICC, RT-PCR, and morphological analysis. First, we want to check that is there is any alteration in the GR receptor present on the plasma membrane. First, we have to check that we have find that there was a significant increase in the total GR receptor which is present in the whole cell lysate. Along with, we have also checked the GR receptor present on the plasma membrane, and in contrast, we are finding that the GR receptor on the plasma membrane was significantly decreased. In the to validate the results of the Western blotting, we have performed the ICC, and we have stained the GR receptor present on the plasma membrane, and we can see clearly that their fluorescent intensity is significantly decreased when the cells were exposed with the dexamethasone 1 micromolar for 24 hours. Next, we want to check that in this stress condition, the sonic hedgehog signaling is altered or not, and we are seeing that the SMO receptor, which is a membrane-bound receptor, was significantly reduced in the whole cell lysate as well as in the plasma membrane. And we have validated our result with ICC, and over here we can clearly see that the membrane receptor was significantly decreased when the cells were exposed with the dexamethasone. Patch and glia are also following the same pattern, that their expression was significantly decreased when the cells was exposed with dexamethasone as compared to the control group. And from here we can speculate that dexamethasone is significantly altered the sonic hedgehog signaling. Next, we want to check that when the co-treatment with pormorphamine, a smoother receptor agonist we have given, then they are going to overcome this deleterious effect of dexamethasone or not. And we can clearly see that in the pormorphamine group on the plasma membrane, along with the dexamethasone, was significantly increasing the effect as compared to the DEXA group. And the results were also in align with the ICC. Next, we want to check that the sonic hedgehog signaling, and over here the SMO receptor is also significantly increased in the Western blotting results in the plasma membrane as well as in the whole cell lysate. And the results are also in align with the ICC, that the fluorescent intensity in the third panel, which is dexamethasone along with the pormorphamine, was significantly increased. When the SMO is activated, that leads to translocation of glee to the nucleus, and over here we can clearly see that in the nuclear fraction there was a significant increase in the glee expression as compared to the dexamethasone only treated group. And we have also performed the same activity with the whole cell lysate, and we are getting the same sort of results. Next, we have checked the expression of BDNF, profilin-1 and SOX2, which are responsible for neuronal structure. And over here we have checked the expression level on both the setup in gene as well as on the protein level. And we are finding that they are following the same pattern, that profilin, BDNF and SOX2 are significantly increased when the cells were treated, co-treated with dexamethasone along with the pormorphamine as compared to the disease group, that is dexamethasone group. Next, we have checked the AKT and GSK signaling, and over here there was a significant increase in the expression level of GSK, and phosphocrab and FOXO3 are also following the same pattern. Next, we have performed the cell proliferation assay, as dexamethasone was deleteriously affecting the cell proliferation, and over here we can see that there was a significant increase in the pormorphamine, increasing the cell proliferation, and the neurite outgrowth is also regained when the cells were treated with the pormorphamine. And from this we can speculate that a deleterious effect of pormorphamine is reversed by the treatment of pormorphamine, and which is leads to the affect the neuronal survival and functioning of the neurons. Thank you.
So there are two types of GR receptors, alpha and beta. So which one did you examine in your studies?
So we haven't go for the bifurcation of alpha beta. I have just checked the GR receptor, total GR.
Serial number 5, right?
Good morning, everyone. I'm Abhishek Sarkar from the Department of Pharmacology and Toxicology, Naipar Mohali, here to present our research work, which is titled as Amelioration of Diabetes-Induced Cognitive Impairment by Inhibition of Hippocampal TRPC5 Channel by Clemizole Elucidation of Underlying Molecular Mechanism. So diabetes causes cognitive impairment by various processes, which mainly involve oxidative stress, insulin resistance, formation of advanced glycation end products, etc. So the mainly available drugs are cholinesterase inhibitors, NMD receptor antagonists, and monoclonal antibodies. However, these drugs only gives the symptomatic relief. So there is need of discovery of new targets. So after intensive literature review, we found that TRPC channel, that is transient receptor canonical channels, they are involved in oxidative processes and cognitive function processes. So from here we found that this TRP channels can be a potent target for this treatment of the cognitive impairment which is induced by diabetes. So we have chosen TRPC5 channels for the same because they are highly expressed in hippocampal area, which is center for this memory and learning process. Now coming to the material and method. So here we have induced type 2 diabetes in male Sprague-Dawley rats. So we have fed with the male Sprague-Dawley rats fed with high-fat diet 2 weeks prior to the induction of diabetes by low dose of streptozotocin, that is 35 mg per kg dose. After that, the blood glucose of the rats were checked, and the blood glucose higher than 250 mg per dl were taken for the consideration, and they were fed with high-fat diet for throughout the 12 week of this study. The drug clemizole, which is a TRPC5 inhibitor, which was administered in 11th and 12th week of the study. So after the 12 week completion of 12 week, so there was assessment of behavioral test by Y-maze and Morris-Water-Maze test, and after that the rats were sacrificed and their hippocampus were taken, and immunoblotting, histopathological, biochemical and metabolic parameters were evaluated. Now, this is the effect of clemizole on metabolic parameters. So we can see that diabetes significantly altered the level of this metabolic parameters. However, there was no significant change due to the clemizole treatment. So it confirms that the drug has no effect on the metabolic parameters, which is altered by the disease. Now coming to the behavioral effect of the clemizole on behavioral parameters. So here, both in Morris-Water-Maze test and Y-Maze test, there is significant declination of the cognitive function. However, due to the administration of clemizole at both 10 and 30 mg dose, it significantly reversed the effect. So we also checked the expression of TRPC5 channel, which was altered due to the diabetes. Here we can see that the expression of TRPC5 channel has been significantly enhanced, which was reversed due to the clemizole administration. Also, the calcineurin A expression was also increased, which signifies that calcium toxicity takes place due to this diabetes condition, which is also reversed due to the administration of HPLs, this clemizole drug. Now, we have also quantified the gene expression of parvalbumin, calsequestrin, and calpain. So these are altered due to the diabetes induction, and the calpain activity was also increased due to the diabetes. However, it got reversed due to the clemizole treatment. The level of inflammatory markers like TNF-alpha, IL-1 beta, and IL-1-6 that also increased in the hippocampus due to diabetes, and it also got reversed. We also checked the effect of diabetes on the memory-related proteins like PHD95, phosphochem-K2, phosphocreb, and PKC-zeta. So the expression of this protein got significantly decreased due to the induction of diabetes, and however the drug has significant effect to reverse the reduction of these memory proteins, so ameliorate the cognitive process and synaptic processes. Now we check the effect of clemizole on mitochondrial biogenesis protein and complex activities. So this biogenetic protein expression like PGC1 alpha and TFAM got significantly decreased, and which was reversed by the clemizole treatment, and same with the complex activity. We can see that the activity got significantly decreased and it again reversed after the treatment. So we also performed the crescile violet staining. Here we can see that the number of viable neurons got significantly decreased in disease group of animals. However, it significantly reversed the number of viable neurons again regenerated here in clemizole treated groups. So the conclusion of this study is that diabetes causes cognitive impairment through metabolic imbalance, TRPC5 overexpression, neuroinflammation, and hippocampal damage. However, 2-week treatment of the clemizole, which restored the learning and memory performance by normalizing TRPC5 channels, calcium homeostasis, reversed the mitochondrial dysfunction. Also, this clemizole intervention reduced the calpain activity as well as the neuroinflammatory cytokines in hippocampus of diabetic rats. Thank you. So, sir, yes, sir. So, we checked this HbA1c and plasma insulin level to validate that it is type 2 diabetes only. However, this model is already established that high-fat diet first causes the insulin resistance within the cell, and after that the streptozotocin treatment, this causes the dysfunction of beta cells of the pancreas that reduce the insulin release in the body, which is the clinical hallmark of type 2 diabetes. So this hyperinsulinemia and this HbA1c level also confirms the same, and also these metabolic markers as well. Sir, we took the dose from literatures. However, the same dose has been effective in Parkinson's treatment as well in previous literatures.
That begs the question. You finally showed the effect in cognitive impairment.
Yes.
Why do you have to go via diabetes to show that? Because there is one study was presented on, I think, brain injury, then earlier Parkinsonism, and you are showing cognitive impairment with the same mechanism of action. Why via diabetes? Because diabetes, cognitive impairment is a very distant complication. It is not really at all clinically important. I think there are more difficult problems such as renal failures, which is causing CKD or other heart organ. Why do you have to go via this route? Clemizole is available. Commercially it is available. उधर भी ऑक्सीडेटिव स्ट्रेस है क्या अलग है इधर Have you correlated with your clinical disease where this diabetes induced cognitive impairment? What is the use of this study? What is the use of this study clinically? आपने किया है नहीं किया बोल दो no problem. Again, I'm saying
neurochemical interplay. As we all know that dementia and epilepsy epilepsy are both of the common neurological disorders nowadays, and there are around 50 million people who are affected by the epilepsy, and 416 million people affected by the Alzheimer's disease globally. But there are the studies who have suggested that when the people are suffering from the epilepsy, there are the chances to develop a dementia within. And in the parallel, the studies has also shown the patients who are suffering from the Alzheimer's disease having a risk to develop the seizures within them. So this shows the bidirectional relationship between the epilepsy and the Alzheimer's disease. Till now, whatever the drugs are present for the Alzheimer's disease as well as the epilepsy, they have been used to treat these comorbidities, but they have shown to the adverse effect with the standard anti-epileptic and anti-Alzheimer's drugs. Thus, there is no such animal model which are present with the face and construct validity showing pathologies of both the diseases in a single animal model, and we are not able to get a common targets for treating both the comorbid condition at the one time. Thus, we have done experimental validation of a model which is going to be show you the pathologies and the neurochemical interplay to find out the common neurochemical targets for managing both the diseases in a common way. So this is a study. We have taken a PTZ model, that is a standard epileptic model, and then we are further proceed with the SZ model. In the parallel, we have run these model from day 1 to 35. The kindling was done, and on the same time, parallelly we have started the SZ model. After the 21 days of SZ administration by the ICV route, we have divided our group into the two group. One is going to be get as a control SZ model, and other group is going to be get a exposure of a PTZ for the kindling development seizures and all. After that, we have done the behavior parameter, novel object, elevator plus maze, radial maze, further sacrificing the animal, and proceed for the biochemical parameter as well as the neurochemical analysis. So result shows when there is a induction of epilepsy, the kindling stage are going to be reached to the generalized tonic-clonic seizures according to the region scale, that is a stage 5. Within the 35 days, total 17 injection was done for the PTZ model. But on the same time, when we have injected the PTZ in the SZ mice, they only took the 12 days, that means 6 injection to get the stage of a 5 that is a generalized tonic-clonic seizures according to the region scales. This has also shown that the onset of seizures has been reduced in the SZ model. That means the mice which is already having an ADS pathology, they have been reduced with the seizure threshold. So this is the graph which shows that when we are at the 6th injection, the PTZ model is only at the stage 2nd according to the region scale. But however, when we reach the kindling in the SZ model, they have been reduced as the stage 5 according to the region scale. So the severity has been increased in the ADS pathologist mice. Biochemical estimation suggests that there is an increase in oxidative stress as evidenced by the GSH and TPAS parameter. Further, they have been also suggest that the acetylcholinesterase enzyme activity has also been increased as the level of acetylcholine is reduced, which is essential for the cognition and memory formation in the central nervous system. Behavior parameter shows that there is a reduction in working memory, reference memory, special memory, and recognition memory as evidenced by the increase in number of mistakes from the RAM, that is radial arm maze test, and then further increase in the decrease in the discrimination index and increase in the time exploring for the elevator plus maze. Neurochemical analysis we have done for the excitatory as well as inhibitory neurochemicals such as glutamate, GABA, glycine, arginine, taurine, and we have also detected the levels of monoamines such as norepinephrine, dopamine, and serotonin, which are essential for the formation and all the thinking process in the central nervous system. So this suggests that in the SZ PTZ mice, which has already been ADS pathology and then further proceed for the kindling parameter, they have been significantly increased the excitation as well as reduce the inhibition action in the central nervous system. That's why the seizure has been triggered in that, which is a common pathology of the epilepsy. So studies also suggest that sometimes, studies also suggest that amyloid beta is also responsible for increasing the excitation in the central nervous system of ADS patients. These collectively suggest that oxidative stress and neurochemical parameters can be play the common pathologies for epilepsy as well as Alzheimer's disease, and they can be target for the management of both the diseases. This studies is ongoing. That's why we have not identified, I'm not able to present whole the parameters. I'm further proceeding for the histological analysis and the other genomics parameters. Thank you very much. Sir, PTZ is a very well-established model as well as the chemoconvulsant models. So other one is there, but we have already done the parameter with the cornea kindlings too, the other epileptic model too. Yes, sir. Sir, actually we have done the SZ by the ICV route, that is 3 mg per kg route, dosed by 3 microliter single unilateral injection we have done. And this has been reported that when we are going to be give the SZ in the ICV route, they are going to be make a insulin receptor damage over there, and that further lead to cause the amyloidogenic pathways. The APP protein are going to be get misfold. This is the reason for making a amyloid deposition in the central nervous system.
Good morning, everyone. Today's my presentation topic is downregulation of histone deacetylase 6 prevent hyperglycemia, dyslipidemia, inflammation, fibrosis, metabolic dysfunction associated steatohepatitis pathogenesis. Now coming to the introduction, MASH is characterized by necroinflammation due to fat accumulation in hepatocyte with inflammation into the hepatocytes. Approximately 38% of the global population affected by MASH. So several lifestyle factors like epigenetic, altered aberrant epigenetic mechanisms, it leads to dyslipidemia, hyperglycemia, inflammation, fibrosis in the liver. So it may be a logical target disease root cause by modulating these epigenetic alterations. So therefore, various studies reported that tibastatin A possesses anti-inflammatory activity, improves insulin resistance, and decreases glucose level and dyslipidemia. Now coming to the experimental design, in my study I have used C57BL6 mice. There are total 16-week study, 12 week of model development via high-fat diet plus 25% fructose in drinking water. After the model confirmation, we have provided a 6-week treatment of tibastatin A, low dose 10 mg and high dose 20 mg per kg. And further, at the end of the study, we have sacrificed the all animals and perform various morphometric parameters like difference in body weight. The level of the body weight level was significantly increased in MASH group as compared to the control group, but the body weight was in trend in both treatment groups. And we found liver to body weight ratio was significantly increased in MASH group as compared to control group. The level was liver to body weight ratio was not significantly decreased, was significantly decreased in both treatment groups. And further we check the effect of TA on hyperglycemia and dyslipidemia. The level of glucose, AUC of IPGET, and insulin and HOMA-IR, the level was significantly increased in MASH group as compared to the control group. After the treatment of both doses of 2Y, the level was significantly decreased as compared to the MASH group. Further, we check the effect of 2Y and dyslipidemia. We perform triglyceride level, cholesterol level, hepatic TG level, LDL cholesterol, or LDL upon HDL cholesterol ratio. The level was significantly increased in MASH group as compared to the control group. After the treatment of both doses of 2Y, the level was significantly reduced as compared to MASH group. And we check the level of LDL cholesterol was significantly decreased in MASH group as compared to the control group. And after the treatment of both doses of 2Y, the level was significantly increased as compared to MASH group. Further, we check the effect of T on liver function test. The level of ALT, ST, and ALP was significantly increased in MASH group as compared to the control group. After the treatment of both doses of 2Y, the level was significantly reduced. And we check the effect of TA in oxidative stress parameter. The level of nitrite MD was significantly increased in MASH group as compared to the control group. After the both doses of 2Y, the level was significantly decreased. And further, we check the activity of SOD and GSS. The level was significantly decreased in MASH group as compared to the control group. After the treatment of both doses of 2Y, the level was significantly increased as compared to MASH group. Now coming to the HND staining of liver tissue, the HND staining of liver tissue in MASH group shows hepatocellular ballooning in hepatocytes with the cytosis, and as compared to the control group shows normal architecture integrity of the liver tissue. After the treatment of both doses of 2Y, restored this cytosis and hepatocellular ballooning. And these histopathological alterations are restore effectively the structural integrity of the liver tissue. And further, we check the effect of TA on molecular level. We check mRNA expression of these genes like HDAC6 activity was significantly increased in mass group as compared to the control group. After the treatment of both dose of TB, the level was the mRNA expression of HDAC6 was significantly reduced. This is the marker of epigenetic mechanism. And further we check the expression of G6PC, SR, EBP1C, and CHR EBP level was significantly increased in mass group as compared to the control group. After the treatment of both doses of TB, the level was significantly reduced. Further, we check the expression of NLRP3, TGA beta. The expression of NLRP3, TGA beta was significantly increased in mass group as compared to the control group. After the treatment of both doses of TB, the level was significantly reduced. Now coming to the conclusion, TA attenuate glucose dysregulation, reduce liver enzymes, elevate oxidative stress, and restored inflammatory and fibrotic gene expression via downregulation of HDAC6 expression. So these findings support that tubistatin is a promising therapeutic candidate for mitigating metabolic and inflammatory disturbances associated with MAS. Thank you. Sir, tubistatin is a HDAC6 inhibitor. Tubistatin is a selective HDAC6 inhibitor. In MAS condition, various epigenetic mechanisms play different significant role. MAS is a metabolic dysfunction associated with NAS, non-alcoholic steatohepatitis. Sir, NAS is a word is like a stigma for the society. That's why the reason is converted. MAS symptoms like obesity, type 2 diabetes, and insulin resistance. Sir, yes, sir. Yes, sir. Sir, because in the disease in MAS, cirrhosis is the first condition, then inflammation. So, sir, based on the high-fat diet model, so that's why the reason we have used. Yes, sir. Sir, here is. Okay, sir.
Any quick comment?
Sir, I have provided the HDAC6 inhibitor, so deacetylation inhibitor, sir. Remove acetyl group from the lysine residues via this compound. Yes, sir. Yes, sir. Sir. Yes, sir. Yes, sir. But sir, I have performed mRNA expression of TGA beta is a marker of fibrotic gene. Sir, I will be performing in my lab. PSR staining is the marker of fibrotic staining. Okay, thank you, sir.
Yes, sir. Yes, sir. Good morning, everyone. I am Yoginder. Today, I am here to present my presentation on the topic Visnagin attenuate thiacetamide-induced liver fibrosis by modulating TLR4 mediated inflammation and downregulation of TGF-beta alpha SMA-driven fibrogenesis. This would be the table of content. So, moving to the introductory section, liver fibrosis is an abnormal wound healing process. It is characterized by the deposition of extracellular matrix deposition. When the initiation of pro-inflammatory cytokines takes place, it initiates the activation of hepatic stellate cells. Hepatic stellate cells are responsible for the excessive production of extracellular matrix, which leads to the fibrosis. Here we use thiacetamide to develop our model in Sprague-Dawley rats. Thiacetamide is a chemical synthetic natural compound which is obtained, which is used in various industries, and thiacetamide upon its bioactivation convert in its reactive metabolites which are responsible for the damage of hepatocyte. Now damaged hepatocyte are responsible for the activation of Kupper cells. Now these Kupper cells release the various cytokines which are responsible for the activation of HSCs. Now activated HSC forms the fibrotic tissue, which are responsible for replacing normal parenchyma of the tissue and make the tissue unfunctional. So the rationale of the study that liver fibrosis is global prevalence 3.3%. Visnagin we use here is a natural phytochemical which obtained from plant Visnaga. It shows various activities like anti-inflammatory, antioxidant, and also has antifibrotic activity in cardiac diseases. So these are number of animal approved from ISC, and I grouped these in 4 group control: thiacetamide 200 mg per kg, thiacetamide plus visnagin 5 mg per kg, thiacetamide plus visnagin 10 mg per kg. So this is the experimental timeline of my study. In control group, I administer vehicle up to 8 weeks, and in rest of the 3 group, I administer thiacetamide 200 mg per kg via IP route on every 3rd day. Along with in both the treatment group, I administered thiacetamide with visnagin 5 mg per kg and 10 mg per kg via IP route on daily basis. On the completion of the study, I sacrifice animal and collect blood and liver for performing of various parameters. So moving to the result section, firstly, I perform morphological parameters, and there is a difference in body weight. The difference in body weight was significantly reduced in thiacetamide group as compared to the control group, while the liver to body weight ratio and liver to table length ratio were significantly increased in thiacetamide treated group, and its ratio was significantly reduced in both the treatment group when we compare it with the toxin group. Now we assess the primary injury liver marker, which are ALT, AST, and ALP, and we found that their level were significantly increased in thiacetamide treated group, and their level were significantly reduced in both the treatment group when we compare it with the disease group. Here we perform the oxidative stress parameter. We done the analysis of MDA nitrite. We found that their level were significantly increased in thiacetamide treated group as compared to the control group. Their level were significantly reduced in both the treatment group. The activity of SOD was significantly reduced in thiacetamide treated group. Its activity was significantly increased in both the treatment groups. We done H&E staining for histological analysis, and we found that in thiacetamide treated group there is a fibrotic bridge around the hepatic lobule, which is represented by the blue arrow, and the red arrow represent the hepatocytes damage. In the treatment group, we can clearly see the improvement in the histological alteration, which is caused by the thiacetamide. We done PSR staining for detection of collagen, and here we analyze the image A by using image J software and calculate the percentage fibrotic area, and we found that its area was significantly increased in thiacetamide treated group as compared to the control group, and its area was significantly reduced in both the treatment group. At molecular level, we done mRNA expression and check the genes responsible for inflammation and apoptosis. We found that the expression of TNF alpha, interleukin 6, and NLRP3 were significantly reduced in thiacetamide group, and its action were reversed in both the treatment group. The expression of TLR4 was significantly increased in thiacetamide treated group, and its expression was significantly reduced in both the treatment group. The expression of caspase 3 was significantly increased in thiacetamide treated group, and its expression was significantly reduced in both the treatment group. We checked the mRNA expression for fibrotic gene also. The level of alpha SMA, col1, A1 was significantly increased in thiacetamide treated group, and its expression was significantly reduced in both the treatment group. The expression of TGF beta was significantly increased in TAA group, and its expression was significantly reduced in visnagin high dose group. The relative expression of MMP9 and TIM were significantly increased in thiacetamide treated group, and its expression was significantly reduced in both the treatment group. In the summary section, I highlight that how visnagin is targeting different pathways which are responsible for inducing liver fibrosis by thiacetamide. In the conclusion, I conclude that altogether visnagin prevents the progression and development of liver fibrosis through downregulation of oxidative stress, inflammation, and fibrotic pathways. Thank you. Yes, sir. Inflammatory marker, sir. Sir, interleukin 6. Sir, 5 markers. Red arrow, yes, sir. Yes, sir. Sir, basically red arrow is representing the damaged hepatocytes. At the damage, there is an infiltration of the cytokines which are responsible for activation of HSCs and then formation of fibrotic tissue. Thank you, sir. Vishnagan, yes, sir. Sir, we switch ourselves to a natural phytochemical constituent which has less. Yes, sir. Sir, we purchased it. Yes. Yes, sir. Ethics committee. Yes, sir. I see the approval number I mentioned.
Today uh, environmental toxicants are the physical or chemical agents which influence the biotic and abiotic component in the environment. Polystyrene nanoplastic is one of the environmental toxicants. Polystyrene nanoplastic is measured less than 100 nanometer in size. These are some sources of polystyrene nanoplastics, like used in biomedical industry, manufacture products like cosmetics, fertilizers, waste products, and irrigation. Now in my study, this is the work plan. First of all, I synthesize the polystyrene nanoplastic through emulsion polymerization reaction. Then my studies for 5 weeks. I give polystyrene nanoplastic for 5 weeks to Wistar rats in the dose of 10 mg per kg. And in the last 2 weeks, I gave Idaravone at 5 and 10 mg per kg orally dose. Then the animal was sacrificed and the blood was withdrawn and kidney was isolated. Then the kidney functions and renal oxidative stress biomarkers are estimated. Then the Western blotting of GRP78, CHOP and Caspase-12 was done. So first of all, I synthesize the polystyrene nanoplastic. First I take the purified styrene and DVB and SGS in a 3-neck flask. Then stir it at 250 rpm for 5 hours at 70 degree. Then add the KPS with nitrogen purging in the 3-neck flask. Then after the 5 hours cool down, then sonicate for 15 minutes, then lyophilize the product, and then the analysis was done through Zeta Sizer, IR and SAM analysis. These are the results. The diameter obtained was 45.82 plus minus 3 nanometers, and the Zeta potential was about 41 millivolt, and these are the SAM and FTIR results. Now, in animal study, polystyrene nanoplastic increases the body weight as well as the kidney weight, whereas the Idaravone in 5 mg per kg and 10 mg per kg decreases the body weight, increases the body weight and kidney weight, whereas creatinine and BUN was increased in polystyrene nanoplastic group and significantly decreased creatinine in Idaravone 5 mg and 10 mg per kg group. After that, oxidative stress and Western blot of GRP78, Caspase-12 and CHOP was done. In the oxidative stress parameters, NO, MDA and GSH analysis was done. NO increases sorry, uh, MDA significantly increases in the polystyrene nanoplastic group, whereas in the treatment group, the level of MDA was decreased significantly, whereas in the GSH polystyrene nanoplastic group, GSH was decreased, and in the treatment group of Idaramon, it significantly increases. Whereas in Western blotting results, we see that the level of CHOP, caspase 12 and GRP78 was significantly increased in polystyrene nanoplastic group, whereas the level was decreased in the treatment, both the treatment groups. So my conclusion is the establishing the role of ER stress in the polystyrene nanoplastic induced nephrotoxicity.
Good morning, respected judges and delegates. So today I will be presenting my work, Development of an Experimental Cardiorenal Syndrome rat model and Evaluation of Cardio-Renoprotective Effect of calcitriol. So in previous literature, we found that vitamin D deficiency is prevalent and highly associated with poor prognosis of pulmonary arterial hypertension. And usually, what happens in pulmonary arterial hypertension is that there is RV dysfunction, which is the major cause of death in these patients. And renal dysfunction is known to be an independent predictor of mortality in patients with both pulmonary arterial hypertension and RV failure. So this complex disorder in which cardiac dysfunction leads to kidney dysfunction is known as cardiorenal syndrome. So our aim was to evaluate the calcitriol effect of calcitriol pre-treatment in a novel experimental model of cardiorenal syndrome in rats. Our model of cardiorenal syndrome was secondary to pulmonary arterial hypertension, which we induced using monoprotein, where there was an increased oxidative stress and inflammation, which leads to an increased apoptosis and proliferation, which leads to a progressive vasoconstriction of pulmonary arteries, which results in an increase in the afterload in the RV. Primarily, we worked on UX cell lines, where we induced the disease signaling through TGF-beta, and we wanted to see the effect of calcitriol on the protein expression of both eNOS and alpha SMA. So what we observed that after giving calcitriol treatment, the eNOS expression was significantly increased and the alpha SMA expression was significantly decreased. And we also observed that after knocking down a VDR receptor, this sort of activity of calcitriol was decreased. Our experimental model was based on two of the drugs, monocrotaline and cisplatin. We gave a single dose of monocrotaline and multiple weekly increasing dose of cisplatin. We started our calcitriol pre-treatment 4 weeks prior of induction of the model, and we gave calcitriol treatment 4 weeks for during the period of induction of the model. This was the mortality in both monocrotaline plus cisplatin group. It was 33.33%. In the RVSP, you could see that group 4, which is monocrotaline plus cisplatin, the RVSP was significantly increased. And this was significantly reduced by calcitriol pre-treatment in the group 6th. Similar sort of trend was seen in RVOT and AOT ratio, that monocrotaline and cisplatin is it because of the VDR ACE2 axis. We also measured this sort of trend in renal injury markers, where in group 4 there is an increase in the skin unit regardless, and this was significantly decreased, as you could see, in the calcitriol pre-treatment group. No problem. In the histopathology, you could see that there is significant which further confirms yes, we actually didn't.
And especially it's important because the vitamin D metabolism impacts.
Did we end up? No, no, madam. When say some madam here, I thought it is the last one. Yeah, please.
We are honored to have Dr. Nilima C. Chagar, is a renowned clinical pharmacologist and former Vice Chancellor of MUHS and former Dean of GSMC and KM Hospital, Mumbai, and a national leader in advancing clinical research, drug regulation and rational medicine use in India. Her contribution have inspired generation of healthcare professionals. Thank you, ma'am, for coming. Please have a big round of applause for her.
That's fine. Yeah, please go ahead. Number 30. Okay.
Good morning to all. Myself Jagdish and I am from BITS Pilani campus and I am working under Professor Pragyanshu Khare. Today I will be talking about hidden target for metabolic disease, which is a peripheral vasomotor tone. So the title of my presentation is targeting. It's okay. Am I audible to all?
Yeah, of course.
Okay. Title of my presentation is targeting adenosine alpha 2b receptor enhancing the heat dissipation and thermal sensitivity, which lead to an improved metabolic.
Please use mic. वो record हो रहा है ना आवाज वहाँ पे इसमें problem है.
Which lead to an improved metabolic profile in mice. As you know, in our daily life, we expose to different environmental temperatures. So, in the 12 in the year, we are faced to hot temperature also, cold environment also, and so in that temperature, the first primary physiological response is peripheral vasoconstriction. Which is why that is peripheral vasoconstriction? Because if we face to cold temperature, first response we face, first physiological response is peripheral vasoconstriction, which is to maintain the core body temperature, which is basically for retaining the heat. Actually, our surface vessels try to that get constricted in cold environment, and our body try to retain the heat to maintain the core body temperature. Similarly, in mice, if you know the normal housing, you already know the normal housing temperature is 22 to 25 degrees Celsius in the laboratory. So, at this as per the concept of thermoneutral zone, which was given by Andrzej A. Rabanovsky in 2002. So, according to his concept, this temperature is not thermoneutral for the animals. And here, at this temperature, there is a subtle sympathetic nervous system activity, which is basically important to regulate the core body temperature and to maintain the energy homeostasis. So, if we expose the animals at cold environment, so at this, the sympathetic nervous system play important role, and there is increase in energy production. There are activation of different pathways. The first is the peripheral vasoconstriction, second non-sibling based thermogenesis, and third there is to meet the energy demand. There is increase in feed intake also and activation of thermogenesis, sibling based thermogenesis pathway. And next, if we expose the animal in hot environment, so at this condition to maintain core body temperature, first the animal try to dissipate the heat. The primary organ is that mostly the animal dissipate heat from the tail, and here in warmer temperature there is loss of energy which is from the surface, so there is vasodilatation in this condition, which lead to increase in heat dissipation and which further maintain responsible for the maintaining the energy homeostasis and responsible for the maintaining of core body temperature. So here in my presentation, we are targeting a unhidden target, which is the adenosine alpha 2b receptor. The reason behind this, actually, this adenosine alpha 2b receptor is basically involved in peripheral vasoconstriction. So here we are targeting this. So we use a pharmacological approach. So here we use a antagonist for this receptor, which is ERC-239. So first we have did an acute study. So in that we checked the effect of this particular antagonist in the mice fed with normal pellet diet, and we performed thermography, and we also did some behavioral parameter tests. And further we did chronic study to validate our acute study experiment. So now coming to the results. So first, in the first experiment we have did IR thermography. So in this we had captured the thermal images before the in the animals treated with this particular antagonist ARC-239 and without the treatment. And we, according to the data, we observed that there is significantly decrease in the peripheral vasoconstriction, which is indicated by the increased blood flow in the tail. So this confirms that this targeting this particular receptor is increasing the heat dissipation from the surface. So as you know, if there is there is increase in the energy loss. So for the compensatory, there will be increase in feed intake also to maintain the energy homeostasis. For confirming the effect on that, we have done the next experiment. We induce hyperphagia in the animals, and so for this experiment, we have housed the animals at two different temperatures. First at 25 degrees Celsius and then 30 degrees Celsius. So 25 is just the normal housing condition, and 30 we choose because 30 is the thermoneutral temperature for the animals. So thermoneutral temperature is the temperature at which the animal require minimum energy to maintain the energy homeostasis, and they dissipate minimum energy to maintain their core body temperature. So here we have first induced hyperphagia. Then according to the data, there is significantly decrease in the feed intake after the treatment of this adrenergic alpha-2 similarly, we also performed glucose tolerance test. According to this data, we also observed that there is significantly increase in the glucose disposal. So, and next we confirm these experiments in the high-fat diet. We have induced metabolic alteration after feeding the C57 mice with high-fat diet. Then we again perform the same experiment to confirm the effect on heat dissipation. So here you can see also the animal treated with this adrenergic alpha-2 receptor, there is significantly increase in the...
You have to conclude, Jagdish.
Okay, sir. And next we have checked the thermal sensitivity for, as you know, in obese object, there is impaired thermal sensitivity. So we have performed this thermal two temperature preference test. According to this also confirms that this particular antagonist is also increasing the thermal sensitivity profile in the animal fed with high-fat diet. And further, we also check the glucose disposal in the animals fed with high-fat diet, and we observed that there is significant reduction in the this blood glucose level, which indicate that it also play a role in glucose homeostasis. And in the conclusion, we conclude that this targeting this adrenergic receptor can be this is the first ever study which we have performed by modulating this peripheral vasomotor tool for the treatment of metabolic disorders. And here this adrenergic alpha-2 antagonist shows a significant decrease in the hyperphagia. And last, I want to acknowledge my supervisor.
That's fine, don't worry. Okay. Now, sorry, you are Jagdish.
Yes, sir.
You have spent more time on background itself.
Actually,
that's
this is
anyhow, that don't worry. It happens. It's a kind of competitive session, you know. We need to keep it in mind that any quick question.
Yeah. So first question. Good presentation. Second is last slide you mentioned metabolic complications. Yes. So what are the metabolic complications that you have the answers in your presentation only? What are the metabolic complications do you think that this can help prevent?
So basically, first obesity. So here we are talking about the normal temperature, that is 20 to 25 degrees
Okay.
Celsius.
What else? What else? Obesity you mentioned. Okay. Number one.
Basically, obesity is a hallmark for other diseases like diabetes and cardiovascular disorders.
Fine. What else? What happened? It reduced the peripheral vasoconstriction.
Yes, sir.
What does it implicate?
So it increases the heat loss from the body.
Clinical implication क्या है उसका?
It will help to treat the metabolic diseases.
Like
obesity. First, obesity.
Hypertension बोलना भाई.
Hypertension also.
Yeah. So that is very important. So metabolic syndrome.
But according to the literature, sir, this if we knock out this particular, so mice also did this working this ADA to be knockout animal.
Okay.
So there will be no change in the blood pressure in that.
Won't it decrease the afterload?
What, sir?
Won't it decrease the afterload?
Yes, it will decrease the afterload.
Yes. So these are again I'm saying related to what is the clinical situation, how it will benefit. Your research work has to benefit the patients. Okay. Thank you.
Thank you.
Mrunali More. morning everyone present over here. I am Ranali More and today I will be presenting my research work on intranasal delivery of lobiglutazone nanoemulsion, a promising therapeutic strategy for Alzheimer's disease. So in this talk, I will be talking about the background, why we have chose this drug and the formulation approach and the detailed results about our animal study. So talking about, as we all know, Alzheimer's disease is a serious illness due to memory loss, and unfortunately, the current medicine are mostly helpful for symptoms, but do not have much effect on the disease are getting worse. Since the new formulation is having an expensive and long lasting time, so we are mainly focusing on repurposing of the drug. So our main aim and the objective is to investigate the therapeutic potential of the lobiglutazone for Alzheimer's disease through in silico in vitro assays, and mainly we are focusing on the nanoemulsion formulation through intranasal delivery. So the rationale behind is that the animals are induced first with the LPS model, that is lipopolysaccharide, and then it will eventually increases neuroinflammation, and then our drug, that is lobiglutazone, which will be induced orally and intranasally, so it will eventually decreases the inflammation. So the material we have used is LPS, that is lipopolysaccharide. Lobiglutazone was procured by Glenmark, and donepezil is used as a standard from Microlab. The methodology I have done is network pharmacology, docking, in vitro assay, formulation, ex vivo study, and preclinical study. Coming on to the network pharmacology, as we have done different database, that is Swiss ADME, binding DB. From that we have got the common genes, that is 34 common genes I have got. Then we have done strings. Then also we have done shiny go. From that we have got different pathways like cellular component, molecular function, and biological process. Then we have constructed a network pharmacology from cytospace. From network pharmacology we have got 10 common protein. So we have whichever we have got the higher degree, we have taken that and we have done the docking of that. So more the negative docking score, it will show stronger affinity. So we have compared with the ligand complex and our drug protein. So next we have done the in vitro assays. In that I have performed RRBC. Here it is shown blue line is representing standard and orange line is representing my plain drug. So higher the membrane stability, it will show good in inflammatory property. Then I have also performed ACH, and likewise I have also performed FRAP assay for antioxidant property. Next is drug profile. I want to say my drug is lobiglutazone. It is an anti-diabetic type 2 drug. It is thiazolidine class. So it is BCS class 2, so it has poor solubility and good permeability. So we are using this drug intranasally rather than orally, because orally it will show poor solubilization and low bioavailability. So we are using intranasally for stability and solubility purpose. Next we have done drug excipient. So we have used different excipient to different batches. So we got capmule oil is showing better solubility with our drug. Then we have performed different batches and we have selected one batch. We have done oil in water emulsion. We have also performed different parameters, particle size, zeta potential, which is comparatively good, showing affinity with the standard values. Then we have performed FTIR. We have also performed DCS, TEM, an ex vivo study for permeability, which is nasal mucosal membrane. It is showing 1.13 fold enhancement. We have compared with the plain drug and our formulation. Next, we have also studied preclinical study. We have taken rat LPS induced model. Each group has 8 animals. This is my study protocol. 7 days I have given LPS, 8 to 14 days I have given drug, standard drug and my test drug. After that I have performed parameters and then sacrifice. These are the behavior study. Elevated plus miss open field test. In elevated plus miss we have performed, we have checked percentage time in the open arm for anxiety purpose, then open field test for locomotion, then novel object for recognition, and water maze to memory check. So after all this I have concluded that intranasally it is showing significant effect. Then oral, this is the antioxidant property. Sir, so I conclude that my intranasal property is showing more effect than the oral. These are my reference, and this is.
Here, you have taken an anti-diabetic,
Yeah.
retazole,
Yeah.
and you wanted to repurpose towards LG.
Yeah.
Okay? In doing so, you have taken oral as well as
Intranasal.
Did you compare with known anti-Alzheimer?
Sir, doneprazil I have taken as a standard.
So, there you have taken as a oral or intranasal?
Sir, both.
Both. But did you get any doneprazil in the market as an intranasal? Is it available?
Sir, it is available.
Intranasal.
Yeah.
Intranasal.
So it is orally it is available, but intranasally it is not available. That's why I am comparing with both. Yeah, yeah.
Sir, one quick comment or question. Madam, is there?
So. Sir, company? हाँ, approval. Yeah, sir, we are trying to apply for the patent. It is in process. So, yeah. Thank you.
Yes, sir. A very good morning to one and all present here. I am Jiten Singh and I am presenting my work entitled 4-phenylbutyric acid attenuated the acrylamide induced testicular toxicity through the ER stress in adult Vista rats. And this work was carried out under the expert guidance of Dr. Ashok Jangra, Dinesh Dhingra sir and Dr. Dinesh Kumar sir. So, what is the acrylamide? So, acrylamide is a processed pollutant that is formed when a carbohydrate or a starch containing foods are processed at the high temperature, that is more than 120 degrees Celsius for a time duration. So, this is occur because of the Maillard reaction, which is occur generally in between of the reducing sugar as well as the asparagine. And as per the WHO data, the average daily intake of acrylamide is about 0.3 to 0.8 microgram per kg per day for the humans, and it is responsible for their toxic effects. But in a study, it was recently reported that the protein-rich food, when processed at the high temperature, that is more than 120 degrees Celsius, they produce almost 5 to 50 microgram of acrylamide. But the carbohydrate-rich food is processed at the same temperature and the same time, they form almost 150 to 4,000 microgram of acrylamide. So these are the some source of exposure. Generally, we take the like potato chips, finger chips, and roasted breads. They all contains the high exposure of high content of acrylamides. And in laboratory, we use the bisacrylamide as well as the acrylamides. Then we just come into the contact of the acrylamide, and it is also used as a flocculant for the sewage water treatment. Okay, sir. So this is my target pathway, the ER stress pathway. The acrylamide is generally known for the induction of the oxidative stress, and we are trying to find their role in the ER stress induction. So this is my work plan, and we design our study, and we divide all the animals into the 4 different groups. The first one is control, then second one is the acrylamide treated rats, and third one is treated with the 4-phenyl butyric acid, which is a well-known ER stress inhibitor. That's why we take this drug at the dose of 500 mg per kg as well as 1000 mg per kg. After the completion of the 28 days of the study, we go for the sexual behavioral analysis and as well as the serum testosterone level analysis. Thereafter, we sacrifice the animal and go for the sperm quality and quantity analysis, biochemical estimation like the oxido nitrostatic stress biomarkers, histopathology, and as well as the Western blot analysis. So these are the results. So in our study, we found that the primary indication of the toxicity is the loss of body weight. So here we clearly found there is the significantly reduction in the like the body weight as well as the testis weight and the epidermal weight, and this is indicating the gonadosomatic index, which is the relative weight of the gonads to the body weight, and it was completely restored, and almost more than 100%. It will that indicates that they promotes the body growth also, sir. So thereafter we go for the sexual behavioral analysis. Here we check the sexual motivational behavior as well as the copulatory behavior. In the sexual motivational behavior, we evaluate the motivation of male rats, how much they motivated for sexual performance. So here we check the time spent. We design a arena and we divide into 2 different parts. We consider this. First one is the male zone and another one is the female zone. So we check the time spent of male rat in their oven zone or in the female zone. So on the basis of this, we calculate the preference score, how much they prefer to stay with the female rat or they stay alone. So there is the significant reduction in the prefer to stay with the female rat. So, and after that treated with our drug, it was significantly restored. So thereafter we check the copulatory behavior also. So here we check the latency to the first intermission as well as the mounting latency, ejaculation latency, intermission frequency, and the mount frequency also, sir. So thereafter we go for the evaluation of oxidonitrostatic stress biomarkers. So here we check the acrylamide induce the oxidative stress in the testicular tissue, and the increased level of nitrite as well as the MDA is indicating there is a significantly increase in the oxidonitrostatic stress biomarker, and the reduced glutathione, the decrease in reduced glutathione level is indicating the alteration of the antioxidant enzymes. So our treatment provided by us, like 4-phenyl butyric acid, is significantly restored this oxidative insult. So thereafter we go for the evaluation of the effect of acrylamide on the sperm as well as on the testosterone. So we found there is the significantly decrease in the sperm count in the epidermals. And thereafter we also check the morphology of the sperm, and here we found there is the head breaking as well as the tail breaking, and it was significantly restored by the treatment provided by us, and there is significantly decrease in serum testosterone level, which was further reversed by the 4-phenyl butyric acid, and it is the treated rat sperm, which indicates the clear sperm head without any alteration. So to confirm this, we checked the epididymal histology, and here we found there is that acrylamide treated rats epidermis having not any sperm in their epidermis, which is the storage portion of the sperm. So thereafter we go for the histopathological analysis of the testes, and here we found there is a red arrow is indicating the multinucleated germ cell, which is the indicating the degeneration of the germ cell, that is the spermatids. So, and this red arrow is indicating the vacuolar formation, and this red star is indicating the loss of interstitial tissue, that is the leading cells. So leading cell is responsible for the synthesis of for the formation of the testosterone. So the loss of this interstitial tissue or the leading cells is indicating the loss of serum testosterone level also. So and these are some other parameters which we measured through the histopathological analysis, and these are some. So thereafter we evaluated the ER stress related proteins expression. So here we found the overexpression of GRP78 as well as caspase-12 and the CHOP. The overexpression of CHOP and GRP78 is indicating the activation of ER stress, while the lower expression of like GRP78 and CHOP is indicating the inhibition of ER stress. So this is my conclusion, that the acrylamide is induced, when germ cells are come under the contact of acrylamide, they may induce the oxidative stress or the mitochondrial damage, which we for the this work is under the process. So this they together causes the ER stress, and in response to this ER stress, CHOP and caspase-12 apoptotic pathway is activated, and they may leads to the DNA damage or the sperm abnormality, or they may cause the germ cell apoptosis. So our treatment for phenyl butyric acid is inhibit this ER stress. So thank you, sir.
This is your Jiten Singh, right?
Last one, sir. Yes, sir.
Acrylamide.
Yes, sir.
What made you to think about this study actually? What you wanted to prove at the end?
Sir, actually we all are comes into the exposure of acrylamide in the daily use, sir. So that's why. And as per the recent report of the state of world population, they found in India, sir, there is a 27.5 million people are suffering from the infertility. And among these, there is a 45% of cases are only because of the male partners. So that's why we are trying to...
What is the inference of nitric oxide
Yes.
measurement?
Nitrite oxide.
NO, you measured NO also.
NO. Nitrite, sir.
So what is the inference?
Sir, nitrite level, na, sir. Yes, sir.
Made NO, that is nitric oxide.
Yes, sir.
Madam is also here.
Yes, sir.
You have to be more pharmacologically vigilant.
Yes, sir. Yes, sir.
What is NO's role?
So actually, the nitrogen species are, if nitrogen species are increased, they may lead to... Okay. Yes, sir. Yes, sir. Okay. Thank you, sir.
Thank you, sir.
Sir, I am, good morning everybody. I am, yeah.
No, 1 level. Level. 1 minute. 1 minute.
Soon you will have to process.
Similar thing.
Yeah.
Okay,
Sir, I am professor of pharmacology in Veterinary University. I am not competing here for any award. I was the last minute entry, first time in IPS. So
That we will decide
no.
whether you are competing.
The thing is, I was the last minute entry, so it was not even accepting online. So I sent by email and they just adjusted me somewhere. Definitely my talk. Okay. So my talk is on general anesthesia. I'm a pharmacologist working in veterinary hospital in Ludhiana. In Ludhiana, we have got India's largest veterinary hospital, and I invite you all, you can bring you any of your sick animals, and you really like that place. So I am working in the field of general anesthesia.
And Dr. Saab, he was always asking every competitor, what is the clinical significance? What is the clinical significance? And my research is basically in the hospital only. I work in the field of pharmacokinetics. I'm really surprised that in IPS there is no session on pharmacokinetics. So it is not a molecular work. I worked in the University of Pittsburgh in nuclear receptors, 20 impact factor, everything. Then I left, came back to India, worked in clinics, started working as a pharmacokinetic. So my presentation here is the influence of isoflurane general anesthesia on the disposition kinetics of ceftriaxone, tazobactam in horses. Now, general anesthesia is an art. There are 4 stages of general anesthesia, 1, 2, 3, 4. Stage 1 and stage 2, they are stages of excitement. So you don't want excitement, you want to bypass those stages. So straight away you take the animal into stage 3, plane 2. So that's the plane where you do surgery. So when you are trying to bypass the stage 1, stage 2, naturally you have to give pre-anesthetics like butorphanol, acepromazine, glycopyrrolate, and say acepromazine, hypotensive drug, it is a phenothiazine dopamine blocker, so it will cause hypotension. So naturally, when there is dilation of the blood vessels, more drug goes from the central circulation from the blood into the tissues. So you expect volume of distribution to increase, you expect dose to increase. Now secondly, you are giving injectable anesthetic. Before the general, you may have seen gases being given to the patients, but gas goes in, goes to the lungs, goes to the blood, saturates the blood, then goes into the brain. So it takes time. Again, that prolongs the stage 1 and stage 2. So you don't want that. So before the gaseous inhalant anesthetic, you are giving injectable anesthetics like ketamine, xylazine. So again, ketamine, NMDA blocker, xylazine, again, hypotensive drug, alpha-2 agonist. So again, that is causing, dilation of the blood vessels. Again, more drug is going from the central circulation into the tissues, so volume of distribution again is increased, so again you expect the dose to increase, but then you are giving isoflurane. Isoflurane is, causing unconsciousness. General anesthesia is the reversible loss of all sensations and consciousness. But general anesthetics, in addition to causing unconsciousness, they are also affecting cardiopulmonary centers in the brain. So the heart rate, cardiac output, blood pressure is decreased. As a result, the blood flow to kidneys is decreased. As a result, excretion of the drugs and also metabolism by liver is decreased. So on one hand, you are pushing more drugs from the central circulation into the peripheral tissues, and you are expecting the dose to volume of distribution to increase. On the other hand, you are decreasing the blood flow to liver and kidneys. You are decreasing metabolism and excretion. So the dose, the plasma levels are maintained for a longer duration at higher concentration. So that decreases the dose. So what is the balance of volume of distribution increase or the decrease in metabolism and excretion? That needs to be seen. Some drugs, the doses are increased in general anesthesia, but for other drugs, the doses are decreased in general anesthesia. So here, one of the drugs which we are using in horses is ceftriaxone, tazobactam. It is a beta-lactam antibiotic combination. And in all the surgeries, we have to give antibiotics because the golden period of wound healing is 5 days. Especially in Indian conditions, you have to give antibiotic at least for 3 days. So you have to give antibiotic. Now you don't know what dose to give because in general anesthesia, the companies, they are providing doses for unanesthetized animals. But here we are using anesthetized animals. So we have got 2 groups. So the objective is to study the differential effect of isoflurane general anesthesia on the PK, and we have given tazobactam, and we have used 5 anesthetized horses. The unanesthetized horses were from the ITBP Panchkula, and 5 horses which are coming to our clinics for general colic surgery, we have used them for the anesthetized group. And you can see the heart rate is clearly decreased in general anesthesia. The blood pressure, there's hypotension in anesthesia, and also respiration rate, depth of respiration, everything is decreased. So I've got just 5-6 slides. Don't worry about time. So, okay. So the plasma concentrations. Okay. So the plasma concentrations. Group 1 is unanesthetized horses. Group 2 is anesthetized horses. So you can clearly see the plasma concentrations anesthetized group, they are higher, significantly higher at all time points. In the unanesthetized group, the drug concentration was detected up to 6 hours, whereas in the anesthetized group, it was detected up to 8 hours. So more concentrations for longer durations are there in the anesthetized group. The volume of distribution is increased, you will see that, but the blood flow to liver and kidneys is decreased to a greater extent. So that is contributing more as compared to the increase in volume of distribution. So these are some of the PK parameters compared in the two groups. This is the unanesthetized, anesthetized group. So you can see the volume of distribution is increased, definitely. But you can see the clearance is clearly decreased. Also, the area under the curve, that is increased, okay? So based on these PK parameters, the conclusion is we have to decrease the dose of ceftriaxone tazobactam in horses by just 2 milligrams. In unanesthetized horses, we have to give 8 milligrams per kg body weight, whereas in anesthetized horses, we have to give 6 milligram per kg body weight, because ceftriaxone tazobactam is a time-dependent antibiotic. It is not the concentration above the MIC which is important, it's the time duration above the MIC which is more important. So we have just minimum T more than MIC is 50, but we are taking say 65. So to maintain percent T more than MIC of 65%, we have to give 8 milligrams per kg in unanesthetized normal horses and 6 milligrams per kg body weight in anesthetized horses. Now 2 milligram per kg difference. The clinical significance which he has been asking is like for a poor cart owner who is having a poor horse, so you have to treat for 3 days at least. So that makes a difference of at least 2,000 rupees over a period of 3 days. So I think that is quite significant. And okay, so my conclusion is isoflurane general. This is the motorized horse. How many of you have worked with a horse? Nobody. How many of you have done horse riding? Nobody. How many of you have touched a horse? Okay, you have done horse riding. Working with a horse is a totally different than working with a mice or a rat. It is at least you need 4 people to anesthetize a horse. So this is a motorized horse, and this is the surgery we are doing. Isoflurane general anesthesia alters the cardiopulmonary parameters, increases the volume of distribution, but decreases the blood supply to liver and kidney by around 40%. So as a result of all this, you have to decrease the dose by about 20%. Okay, thank you very much. Oh, really? Okay. I'll be happy to take any questions, and I'm really happy that somebody has been asking clinical questions.
No, this is significant for the patients. Can be translated to the patient situation in anesthesia. So probably this for I understand that when it goes to human clinical, it could be translated as well.
Now we'll begin with our online session. It's my pleasure to introduce Dr. Yao Yu Yen she's from Sunway University in Malaysia. Professor Yu is an algae biotechnologist focused on discovering new algae-derived compounds for treating neurodegenerative diseases. She also studies their cosmeceutical, antimicrobial, and anti-cancer potential along with algae genetics. There are about 386 algae taxa recorded in Malaysia, many showing strong medicinal properties. Her work supports Sunway University's missions towards UN Sustainable Development Goals by exploring how algae can improve health and well-being. So with great respect, I will welcome Dr. Yu to share her insights on the drug discovery of algae compounds in neuroprotection and skin health.
Hi everyone, I'm Yao from Sunway University, Malaysia. So first of all, I would like to thank Indian Pharmacological Society for inviting me to deliver this guest lecture. The title that I'm going to talk about today is Seaweed Solutions, Neuroprotections, Antioxidants and Skin Health from Bredina australis, Bridging Natural Compounds and Pharmaceutical Innovations. And now I'm going to give you a very brief introduction about algae. So algae, some of you may call them algae, and also some of you may call them as seaweeds. In Chinese, we call them as zao or hai zao, right? So where can you find algae? Algae you can find everywhere. You can find in the swimming pool, in the stream, river, even in the aquarium tank, right? And they can also be found in the Arctic or Antarctic regions. Algae, this is a very big umbrella term, and under it, we can categorize algae into two main groups, which is microalgae and also macroalgae. Microalgae, they can be unicellular or multicellular, and they are microscopic organisms, means that you cannot observe them with our naked eye, but you can only observe them under the microscope, right? So this is how they looks like under the microscope. Okay, they are growing in the media, and this is the flask to show you that you can grow that in the lab base with the bioreactor, or this is another type of we call it raised wave pond. All right, this is another way to cultivate the microalgae. So for macroalgae, this is like plant-like organism. They are multicellular. You can also call them, it's also known as seaweeds. All right. So for microalgae, we can categorize them into three main groups based on their pigmentations, which are red algae, rotophyta, green algae, chlorophyta, and also phaeophyta, which is the brown algae. Okay, and now may I ask you, do you think seaweed is a plant? All right. The answer is no. Why seaweed is not a plant, but it looks like plant? Okay, because these are three main groups that I mentioned to you just now, the seaweeds. Because they are flowers, they do not have flower, they do not have seeds. They are non-embryo organisms, non-vascular plant, and lacking of true roots, stem, or leaf. And then all the cells, they are containing chlorophyll A for sure, because they are going to survive with the photosynthesis process, right? All right, so this is why we call it the thallus. It's not leaf. And then they do not have the roots. They just have a whole fuzz to allow them to attach on the hard surface so that they can attach on it and grow. That's why they are under the Protestant kingdom here. Come to your mind when we talk about algae. Some of you might think about food and beverage, right? Like this kind of snack is very famous. It's from Thailand. Okay, you can, and this is the kombu soup. Okay, or sushi. And for cosmetic product, they also can be used in cosmetic product, for example, face masks. and then as a moisturizer or cleanser, and even in the nutritional supplement, right? And our life depends on algae. So here you can refer to this diagram. It shows the human food chain. Algae is the producer and always at the bottom of the food chain, right? So this algae, and then when the stream feed on algae, they will become the primary consumer, then followed by the secondary consumer, which is tuna. And when human feed on tuna, then we will become the tertiary consumers. So that's why algae is always the producer in the food chain. Okay, we know that our ocean covers 71% of the Earth's surface. So the ocean also produces over half of the world's oxygen and store 50 times more carbon dioxide than our atmosphere. Means that algae can help to clean the atmosphere by capturing the greenhouse gases like CO2. And then because algae itself, they can help to convert the inorganic compounds such as carbon dioxide and water into organic compound with the energy from photosynthesis, all right, from the sunlight. So what they're going to do here is this algae, they will capture the carbon dioxide with the help of the carbonic anhydrase in the cell, and then they will convert it into different biomasses. Biomass, for example, fatty acid, fibers, mineral, protein, okay, and also antioxidant as well. Why algae they are so special? First of all, for their cultivation, they do not require arable land and fertilizing, no weeding and fresh water for their cultivations. Here are the methods usually farmers will use to cultivate them in the open sea. They have rapid growth rate. Some of them can grow almost 6 inches per day. And then they can survive in a harsh condition. Try to imagine this is a seaweed. They are growing in the intertidal zones. In their daily life, what they're going to do is they will use to expose to the UV radiations, and then changes in the temperature and also salinity of the water, the wave actions, and also desiccations during the low tide. So when desiccation happens, the seaweed itself, they will lose about 90% of their cellular water content and can ultimately cause the oxidative stress. So these algae, they are very clever. They have a very diverse mechanism to protect themselves from these harsh conditions. So they will produce secondary metabolites in this case to protect them. For example, like antioxidant molecule and antioxidant enzyme. That's why marine algae or seaweeds, they are rich source of structurally diverse bioactive compound. For example, like polysaccharide, vitamins, lipid, terpenoid, and polyphenols. And some of them, you know, the metabolites from algae are 10 times greater in the diversity than the terrestrial plant. And most of them, they are potential bioactive compounds which are exclusive to algae. Now let's talk about the biodiversity of seaweed in Malaysia. In Malaysia, we have 386 specific and intraspecific taxa. Then in Sabah, so we have Carpophycus species, or you can call them as seabird nests. Okay. And in Kedah, we also have the cultivation for Gassidaria maliniensis, which is the red algae. Another one in Johor, also Gassidaria species. So here are some of the photos that I took when I went for the sampling trip for the seaweed collections. Right, they are very colorful. Yeah, this is very huge in size. This is a sargassum species. All right, and I want to emphasize, bring your attention to this photo at the middle here. So you can see even like one spot we can find there are a number of species of algae available, like green algae and brown algae. Different type of brown algae also available here. So this picture I took in Port Dickson. Okay. Hey, do you know that Malaysia is the 7th largest seaweed producer worldwide? And the annual revenue is over USD 40 million. So here are the Garcia species and Carpophyphicus species that I mentioned to you just now. So Garcilaria species, usually you can eat them as a raw salad. You just have to put some dressing on it, will do. And then for Carpophycus species, usually they will use it to prepare dessert. What they're going to do is you just have to boil this Carpophycus species with the longan and goji berry, alright, which is my favorite dessert. And potential application, for sure you can use them for food consumption, for phycocolloids productions, because they will produce a very high quality of agar and agarose with a very good gel strength, and as a source for the biopharmaceutical product as well. Algae is a sustainable diet as well, because they with a very low environmental impact, and they contribute to the food and nutrition security and a healthy life. They can also help to guide the gut microbes to prolong the human lifespan. Okay, how it works. So as you can see from here, algae, it contains a lot of fibers, right? We can use algae as a prebiotics, where these prebiotics, okay, because algae itself, they have a very complex sugar. This complex sugar cannot be digested by our enzymes in the stomach. Alright, so what you're going to do is, after you consume them, they will be fermented in the large intestine by the microbes. So after the fermentations, they will produce something called organic acid or short-chain fatty acid and gases. And this short-chain active acid, they will help to nourish and also modify the bacterial consortium in our large intestine. Functions of algae in our daily life. So as I mentioned to you just now, algae, you can use them as food or in the beverage, right? You can also use them as food additives and ingredients, nutraceutical and pharmaceutical, in vitamins as well in the cosmetic products. But at the same time, they also can be used to produce bioplastic, animal feeds, to produce the agar or agarose for the research purpose, for water treatment, the wastewater treatment, and also bioelectricity as well. They have a lot, the application very diverse. Okay, now I would like to share with you algae research at Sunway University. So in my research team, we are working on the nutraceutical and pharmaceutical of algae in terms of their neuroprotective activity, their neuroregenerative potential. This is mainly focused on the peripheral nervous system. And then we're also studying their protective effect against the particulate matters induced airway inflammatory responses, their prebiotic potentials, and also their protective effect against the in vivo bisphenol A induced adverse reproductive abnormalities. At the same time, we also study their antimicrobial activity and also their cosmeceutical potential as well. Yeah, we also work on bioplastic. Anyway, today I'm just focused on the neuroprotective effect of seaweeds. So neuroprotection, you can define them as the therapeutic agent which can help to prevent neuronal death by intervening in and attenuating the pathogenesis that result in cellular dysfunction and eventual death. So during neuroinflammation happens, it can cause oxidative damage, and eventually they can lead to the dementia and also depressions. So here are some important findings from our team. So we found that Garcilalia maliensis, they can help to promote growth and development of the nutrients, and also they have the cholinesterase inhibitory activity as well. Here are the publications that we have published. And Campelophycus maliniensis, this is another red algae. We've proved that they have the anti-neuroinflammatory activity. Followed by this is the brown algae, and also what I'm going to focus later in a couple of slides later, Badina australis. So we prove that they can help to treat the depressions and mood-related disorder, and also they have the anti-neuroinflammatory activity. So this is spirulina, which is a microalgae with the anti-neuroinflammatory activity as well. On Badina australis. So Badina australis, this is how it looks like when it collected from the sampling site. After we collect them from the sampling site, we will have to do some processing, like cleaning process, to wash the algae, to remove all the epiphytes. Okay. After this, we will dry them up and then to prepare the herbarium sample. So this is how it looks like, the herbarium voucher. And these seaweeds, Badina australis, usually they distribute in the tropical to subtropical water and in the lower intertidal to deep subtidal zones. And they used to grow on the coral rubbles or reef flats. This slide is to show you the geographical distribution of Badina australis. And then here to show you the biological activities of Badina australis in terms of their antioxidant, anti-neuroinflammatory, anti-inflammatory, antimicrobial, anticholinesterase, neurotrophic, neuroprotections, intestinal immunology, and also anti-cancer. So the chemical constituent of Badina australis, based on the previous study, you can see that they have a lot of chemical constituents have been isolated from these seaweeds. But anyway, in terms of their biological activity, remain unexplored. So that's why we are very interested to further explore about their anti-neuroinflammatory activity and also antidepressants activity from Badina australis. So from here, we have published 2 papers, and this paper we got the Mahathir Science Award Fund as well. So from here, I'm going to talk about the result of these 2 papers. So this is just to briefly show you the methodology, how we prepare the Badina australis extract. So after we collect the Badina australis from Potixons, we do the fish drying and also grinding the Badina australis for the crude extract preparations. Here we are using the sequential methods of extraction with the hexene, ethyl acetate, and then followed by ethanol. And then we also run to study their phytochemical assay and antioxidant assay. So after this, we explore their neuroprotective potentials and also isolate the most potent Badina australis extract for the subsequent assay. So from here, we use it to study the neuroprotective effect using the PC12 cell line, which is mimicking the effect of depressions. Another study is focused on the neuroprotective effect of the extract against the LPS stimulated neuroinflammation in the BV2 microglia cell, and then followed by the isolations. From here, let's talk about the first model, which is the LPS stimulated neuroinflammatory model using the BV2 microglia cell. So what we expect is usually when the cell exposed to the LPS, this LPS, the lipopolysaccharide, they will activate the toll-like receptor 4. So after the activations, what will happen is they will activate, the activations will trigger the protein kinase signaling pathway. Then they will leading to the productions of the ROS and also the NF-kappa B. So the activated NF-kappa B here, they will translocate into the nucleus Then they will promote the expressions of genes such as the COX-2 and also INOX. And then they will result in the pro-inflammatory mediator and also pro-inflammatory cytokine productions. Then finally, they will cause the neuroinflammations. Another one is the high-dose corticosterone mediator oxidative damage model with the PC12 cell. So what will happen here is this high-dose corticosterone, they will induce and increase the intracellular ROS and also ROS in the mitochondria, which will result in the reductions of the MMP and also the aconidase glutathione. But at the same time, they will increase the ER stress, apoptosis, and also LDH. And with all together, they will result in the oxidative damage. Okay, so this is mimicking the effect of depressions. Alright, let's focus on the LPS stimulated neuroinflammatory model of BB2 microglia cell. So from here, we notice that with the presence of the Bedina australis derived antioxidant, it will help to reduce the ROS and also the activations of the protein kinase signaling pathway by reduce the activations of the NF-kappa B and also subsequently reduce the productions of the new expressions of the neuroinflammatory mediator and cytokine. So this is very important because in here they were leading to the suppressions of the neuroinflammations. All right. So for model here, another model, some model here is for the high dose corticosterone mediated oxidative damage model for the PC12. We notice that with the presence of the antioxidants from the Bedina australis, the conditions in fact reverse by 0.25 to 0.5 milligram per ml of the ethanol extract, where with the antioxidant they will reduce the ROS and also at the same time they will help to increase the MMP, aconidase, and glutathiones. At the same time, they will reduce the this is the endoplasmic reticulum stress, apoptosis, and LDH release, so resulting in mitigations of antioxidant damage. Okay, so from here we further prepare the fractionation from the ethanol extract, and then we send the fractions for the LCMS study, and from there the analysis shown that there are 5 compounds, right? 2 esters, and then we have the stearolamide, linoleic acids, and also niacinamides. Based on the 5 compounds that I mentioned to you just now, we also try to explore their reported activity. So from here you can see the linoleic acids and niacinamides. Both of them have been reported with anti-inflammatory, anti-neuroinflammatory, and also neuroprotective effect. So that's why we would conclude that the neuroprotective effect from the Bedina australis can be attributed to these 2 major compounds. So at the same time, we also explore their cosmeceutical potential because algae, they are rich in active ingredient, right? Especially antioxidants compound. So you can use them for the cosmetic industry where the application in terms of their as a photoprotective, anti-aging or skin whitening or for hair care product, right? And in 2020, we published 1 review on algae metabolites in cosmeceuticals. Yeah, this paper is 100 most cited papers in marine drugs. Here are some of the commercially available algae-based cosmetic product in the market. For example, Biotherm, yeah, with anti-aging cream, with red algae, and then Bioessence. This is the moisturizing gel. All right, they consist of 4 types of the algae. And then sunscreen, yeah, we have the geranium species and face mask by L'Oreal. They're using red algae in their product. So based on our finding on the UVB radiation protective effect of Pedaina australis, for this study we are using the HACA cell. So we found that the ethanol extract of Pedaina australis, they serve as a promising source of antioxidants. In terms of the UVB radiation protective effect, both ethanol and water extracts, they will protect the HACA cell from the UV-induced cell damage via promoting the cell viability at 400 microgram per ml. So in conclusion, algae is a next generation functional food and therapeutic agent. Because algae, they are rich in antioxidant metabolites and dietary fiber, and also they can enhance the antioxidant defenses and also gut microbiota richness. So because of this, they will help us in reducing the aging process by reducing the accumulations of the aberrant proteins, reducing the organelle dysfunctions, and also age-related disease. And ultimately, they will help us to promote health and longevity. And currently, we have the cell culture facility in Sumner University. So if you are interested in any algae-related project, yeah, please feel free to contact me. So here are my team members, and also thanks for all the collaborators. Thank you very much for your attention, and nice meeting you guys virtually. So feel free to contact me. My email address is over here. Thank you.
We'll be starting shortly. Dr. Manoj Aswar is on the way.
Manish sir. Or Manish sir can become the speaker.
Yeah, good afternoon to all our distinguished delegates, esteemed faculty members, respected chairs, honored guests, and dear researchers. It is with great respect and profound sense of occasion that we are gathered here for Symposia 29, devoted to the theme of progress, challenges in translational neuropharmacology. The esteemed session is under the distinguished chairmanship of Dr. Manoj Aswar. Now I'll request Dr. Manish to please introduce our first speaker.
Thank you, Jatinder. So our first speaker is Dr. Milind Parle. He is a Dean of Faculty of Medical Sciences at Guru Jambeshwar University of Science and Technology, Hisar. He has over 4 years of experience in teaching, research, and academic leadership. His work spans neuropharmacology, behavioral studies, cognition, and memory disorders. With more than 150 publications and contribution to patents, he continues to bridge traditional medicine with modern drug discovery and serves as an expert to regulatory bodies such as Pharmacy Council of India. I now welcome Dr. Milind Parle to share his insights on yes, I am mentally retarded, but is that my fault? The stage is all yours, sir.
Thank you. Thank you for Then and she said, my son, you are brilliant. You are a genius. You are so sharp that school doesn't have enough facilities to teach you. There are no good teachers to teach you. So, I am going to teach you. Your teachers will not teach you. So sharp and gifted you are. बीच बीच में बंद हो रहा है. Okay. So, the mother expired. You know, this boy became a great scientist. He had more than 1,000 patents on his name, 1,000 patents. You can imagine. Scientist of the century. His name was Edison, Thomas Alva Edison. And then one day he was looking. I'll hold on. Fine. So, after becoming, you know, a great scientist of the century, this boy one day opened the trunk of his old mother, who is no more, late, and in the purse, paper was there. He read the paper. He started weeping. You know what was written in the paper? The paper में लिखा था, मैडम आपका बेटा idiot है, stupid है, we are expelling from the school. He is not fit, he is disturbing the others. And you please take him away. We don't want him in the school. He is retarded. He is mentally deficient. ये सब पेपर में लिखा था. Remembered the words of a great, the words of his mother. Son, you are a brilliant boy. You are so qualified. We don't have enough teachers to teach. So, your mother will teach you. Imagine the change, the transformation made by words of a mother who believed in her son. So much faith. The scientist of the year, Edison, was expelled once upon a time from the school. Next slide. So these are the communication disorders. See, we have various types of them. Cerebral palsy, for example. Cerebral relates to brain, cerebral. Palsy, muscles. When there is a weak movement of muscles, muscles are weak. When you are not, it is cerebral palsy. Then we have autism spectrum disorder. For 100 years, nobody knew. Even when I was doing my MPharm or PhD, it is. We did not. Now, this occurs in infancy, in toddlers, occupational disability, and there are we find various mental disorders. Official books are DSM, published by American Psychiatric Society. Other is ICD. Recent edition of ICD. Indian medical fraternity was unaware for long. Even the doctors, pediatricians did not know what autism is. You can child. There was a child in Bangalore by name Tito Mukhopadhyay, 11 years old child, and this child suffered from child disorder. Mother of this child took in India of this autistic child, and he wrote a book. He could type on typewriter, and he dictated. He could not speak, so he would move his fingers. If he needed food, towards F, O, O, D, meaning I need food. He wrote a book. How such children, retarded children, should be nurtured at home, was taught to the world by a mother of an autistic child of Tito Mukhopadhyay. Tito's mother's approach made it developmental disorder, then autism spectrum disorder in 2013, in 19... Thank you. I hope this is a better mic. In 1922, in 2022, recently, definition has come of it is a spectrum of disorders and lot of improvements in that. The prevalence is very high all over the world. In India, in every 8 families, you will find one retarded child. So high is the prevalence. देखो, जब आप खुद को तराशने लग जाते हो, तब हर शख्सियत आपको तलाशने लग जाती है. You may be a normal child with high IQ, but if you are outdated, if you don't keep yourself up to date, you are like a retarded child. You will be rejected. Now, what are the risk factors? I am answering the question that I am a retarded child. Is it my fault? These are the risk factors. Adverse environment, age of parents. These days, you know, there is a trend. Not to get married is the first thought, and to get married at a very late age. You know, we have a physiology, we have a reproductive system, hormones. There is a age span. Ideal age span is between 19 years to 32 years. This is the highest fertility period. Beyond that, there is a risk of developing childhood disorder. Comorbidities, prenatal, perinatal, postnatal factors. I'll just define this. Prenatal is soon after conception till delivery is period of perinatal, very sensitive. During delivery, there could be lack of oxygen, asphyxia, you know, cesarean, then postnatal, after delivery. So, at any time, this disorder could develop. Short interpregnancy spacing. See, after first delivery, the mother is most susceptible to develop a second delivery. The body is ready. So, if there is a short interval, there is a chance of developing a disorder. Maternal stress, improper diet, alcohol, smoking, these are the risk factors which lead to childhood disorders. Adverse environment. By environment, I mean one is outside environment, pollution, infections. The other is inside environment, uterine environment. Then genetic makeup. See, these are not the causative factors, but these are the precipitating factors. Genes are not in our hands, but environment is in our hands. So genes plus adverse environment, synergistically or laterally, they cause autism. And then neurotoxicant exposure to during sensitive periods of pregnancy lead to autism. Genetics, yes, the science has developed. We have identified genes, 400 weakly associated and 200 strongly associated. These are the genes which are responsible, which could cause the autism spectrum disorders. These are the genes. But we have good news, For a pregnant lady, there are two kinds of factors. We have risk factors and we have protective factors also. So let us focus, find out which are these. The protective factors are optimal parental age, 20 years to 32 years, optimal age. Favorable environment, uterine environment and outside the body environment. Adequate interpregnancy spacing, positive family backdrop. Positive family backdrop. Hindustan mein, yes, we are lucky that way, that we have a sound family system. Though it's declining now, but makes, lady a comfortable atmosphere. Right, I just need 2-3 minutes more. So interplay of protective and risk factors linked to autism. This was published. See, in life, we have two options. Either you accept the things that happen to you, or the first option is you make the things happen. I believe in the first. You either accept the things that happen to you, or you make the things happen. These are the symptoms of autism: deficits in social and professional skills, and defective communication, reciprocal communication. Management of autism. In brief, we have no FDA approved medicine today. What we are addressing is only comorbidities and the symptoms. Drug therapy, serotonin uptake inhibitors, etc. They are all attacking the comorbidities or associated symptoms, not the disease in itself. Yes, non-drug therapy helps. We have complementary therapy as well as nutrition therapy. Like we have cognitive behavioral therapy, speech therapy, music therapy, etc. Strategic management of autism spectrum disorder, another paper we published in a reputed journal. We got is an exemplary article. Dekho, we should not procrastinate. This is a message to youngsters. जिसने कहा आज नहीं कल बस दिन गया टल, जिसने कहा आज नहीं कल बस दिन गया टल, जिसने कहा परसों मान लो बीत गए बरसों, जिसने कहा परसों मान लो बीत गए बरसों, और जिसने कहा आज उसने किया राज. We have a lot of international organizations taking care of autism. There are a lot of Indian organizations. Socially, a lot of funds are available from government. We have, see, autism is a child to be loved. He needs to be respected. It is none of his fault that he is retarded. It's the fault of parents. It's the fault of society, not his fault. Let's keep this in mind. These are some movies, Hindi movies, most of you. My name is Khan. Koi Mil Gaya, Barfi, Tare Zami Par. This theme of autism has been very effectively marketed. In Marathi, we have Math, Yellow. This student worked with me in UIPS Punjab University, Chandigarh. See, we know today much more than what we did many years ago. Nonetheless, we still have plenty to learn and a long journey to travel. This is the team. With Dr. Anil Kumar, we have a DST project on autism. See, if you believe in yourself, if you believe you can, you are halfway there. If you feel you cannot, you are right, you will not. If you believe you cannot, you are right, you will not. But if you believe you can, you can. This is a photograph. You may not identify the student, but I need not introduce you to a great mentor, teacher, who is around us, with us in this conference, legacy of UIPS. Can you identify the teacher? Anyone? Anyone? He is amongst you today, now? Yes, please. Yes, my revered guide. Whatever I am today is because of him. Thank you. Thank you so much. Anybody, if you have any questions, you are free. I am available here for next 24 hours, and I am with you. I'll also give you my email on this topic or any topic, whatever, you know. I'd like to support you. I would like to help you. You're most welcome. Thank you. Thank you for patient listening.
Thank you, Dr. Parley, for your insightful and deeply moving presentations. Your perspective sheds essential light on the area that demands empathy, scientific clarity, and continued advocacy. We sincerely appreciate your valuable contributions to today's program. I will introduce our chair, Dr. Manoj Kashinath Aswar. He is a distinguished pharmacologist with over 20 years of expertise in teaching, research, and mentoring. He has published over 30 scientific papers, delivered 35 presentations, guided 18 MPharm scholars, and led 3 major research projects. As an award-winning researcher and author of recognized pharmacology textbook, he continues to contribute significantly to academic excellence and translational research. I will request him to introduce our speakers. Next speaker.
Our next speaker for today's session is Dr. Vivek Jain. He is the assistant professor at Mohanlal Sukhadia University, Udaipur, with a huge experience of 70 years in neuropharmacology and drug discovery. He has secured 10 research grants, including major grants from ISN, IBRO, totaling around 1.7 CR, with total 40 plus publications, 3 patents, and recognitions such as Young Scientist Award 3 times, 2018, 2023, and 2025. His work continues to advance research infrastructure, innovation, and neuroscience education. I now welcome Dr. Vivek Jain to share his insights on the title From Molecule to Memory, Evaluating Purpurin's Neuroprotective Role in Alzheimer's Disease. Please, sir.
Thank you for a nice introduction, sir. And I am very thankful to all UIPS team, particularly Professor Anil Kumar sir and all the entire team. And I'm very fortunate that I'm first time I'm giving my talk in front of Professor Kulkarni. It is a big time for me, as well as Professor Parle sir. I am also a student of Nirmal sir, and my sir is a student of Parle sir. So that it is also a big time for me. So I'm just starting. We have started our work. We are searching for a molecule that can help in the improve in the memory. So we started like most of the molecule are very costly. So we try to search the natural products which are less costly, so that whenever we translate in the drug, that can be help for the people who are not having much more amount. So my topic is from molecule to memory, evaluating purpurine neuroprotective role in Alzheimer disease. So Alzheimer disease, basically we all know that it is an age-associated neurodegenerative disorder of the brain, which commonly characterized by progressive destruction of memory and cognitive function. However, there are some drugs which are approved by US FDA, and as well as also doctors are prescribing nowadays, like donepezil and some of the NMDA receptor. However, NMDA inhibitors slow down the progression in modulating the target site and provide symptomatic relief. Recently, one of the drug, aducanumab, was licensed in 2021, but it has a more adverse event such as cerebral edema and microhemorrhage are the major concerns. So most of the people are nowadays finding new drugs that which have a natural origin and less side effects. So we are targeting those things. Now, if we are looking on the Alzheimer's, then what we find, around 2019, 55 million people estimated growth in number of dementia. 2030, 78 millions, and if you are looking on 2050, then it will reach up to the 139 million. Then, if you are looking the data, it is every 3 seconds someone in the world develops the dementia, and the total estimated annual worldwide cost of dementia is over US 1.3 trillion. That will also increase up to 2.8 trillion. And this is the map of the Alzheimer's, that it is progressively increasing day by day, and there is a daily associated life burden, people which are living with the Alzheimer's. So in India particularly, it is increasing more and more. And the Peru and the other country which has some less, they are decreasing the Alzheimer burden, but in India there is an increase in the Alzheimer burden. So the pink color showing the higher side, and the dark blue color showing the lesser side. Then there are some factors which affect the Alzheimer's, like physical inactivity, smoking, excessive alcohol consumptions, air pollution, head injury, and infrequent social contact, less education, obesity, hypertension, diabetes, depression, hearing impairment, high LDL cholesterol, and vision loss. These are some of the factors which can increase the Alzheimer kind of memory loss. And then what are the symptoms which we face there is a memory loss, and there is a daily difficulty in performing familiar tasks, problem with the language, as well as disorientation to time and place, and poor and decreased judgment in the case of the Alzheimer. These are some kind of symptoms. Problem keeping track of things and misplacing the things. Generally we find these things. And changes in the mood and behavior, challenges understanding the visual and septal information, and withdrawal from the social activity. So what are the various changes if you are looking on the pathobiology of the IED? There is a mitochondrial dysfunction, tau hyperphosphorylation and amyloid plaque formation, cholinergic insufficiency. Most of the drugs are such in cholinergic insufficiency, and approved drugs are also in this category. There are also hormonal imbalance and oxidative stress and neuroinflammation. These are the some reason behind the development of the Alzheimer's. Now, what are our target? We target the cholinergic deficit as well as decrease amyloid beta toxicity and oxidative stress and neuroinflammation. Generally, in most of the cases, people are targeting a single target, but if we target a multi-target, then there is some kind of possibility that we will achieve our goal. So we target these four things so that if cholinergic and cholinergic deficit decrease amyloid beta toxicity, it will help us. So this is about the gamma secretase and how gamma secretase, beta secretase work, and then further formation of amyloid beta. And this is about the oxidative stress, which further lead to the inflammation and cause the formation of So epigallocatechin 3 gallate is a standard compound for amyloid beta. So we compare with that, and these are the binding results of the epigallocatechin 3 gallate. Then what we did with the in vitro approach, we complete this in vitro approach through the cell culture studies, and we take SHSY5Y cell line used for the cell culture studies and perform the toxicity through amyloid beta 2535. Then just look at the cell survival. So there is a decrease, through amyloid beta there is a neurotoxicity, and various doses of purpurine, it increase purpurine 8 micromolar has a good number of percentage cell survival. So this is the rescue effect of different doses of purpurine on the neurotoxicity caused by amyloid beta SHSY5Y cells on for 74 hours. Then further we also, in the cell line study, we also perform the catalase, T-bars and SOD. So in the catalase, amyloid beta STZ group. In the cerebellum, the normal
Thank you, Dr. Chan, for your enlightening and impactful presentation. Your research offers meaningful advancement in understanding neuroprotection and highlights the promising direction for Alzheimer therapeutics. We sincerely appreciate your valuable contributions to today's scientific forum. Now I will request Dr. Asura to introduce our last speaker, Dr.
Last speaker for this is Dr. Professor Chandigarh. Her research spans gastrointestinal, reproductive pharmacology, significantly to the academic and research.
I want to thank the chairperson, respected chairpersons, to introduce myself. So, Actually, my one of the research area is neuroscience. I am working on this the last. So I faces lots of challenges and limitations. Studies in case of animal model. So our highly focused major challenge to enter into the central nervous system. So how we can overcome that? Now the emerging solution is the nanotherapy. There are various forms of nanomaterial, nano carrier, nano formulations are now trying to give drug. So nanoneuroimaging is an emerging field of precision brain therapeutics. Then translational barrier I already mentioned. Challenges is like directly it is very, very difficult translating the preclinical results to clinical efficacy studies because animal model is lack of predictive validity. The models are not actually like the mechanism which are seen in the patient. So all these are the because majority of these diseases pathology to see is very genetic. So you cannot nowadays genetic model of genetic animals are available, but it is divided out because it's very costly. One genetic animal from Jackson Lab, so how much is cost? So country like India, even in Fiji, are struggling to get genetic models, establish some role of some drug. So translational value definitely. Then our additional barrier in case of is that regulatory hurdles and complex trial design, unclear endpoints, high cost, and obviously to see some effect, you have to keep the drug for long patients. So that is one of the barriers. Then challenges barrier besides this is regarding the clinical trials, mainly the patient population. So if you see in the published literature, any of the published journal studies the neurological disease is very limited or very less as compared to studies conducted in cardiovascular disease in other area of therapeutic area because we have little understanding about the disease and brain is there is no innovation actually happening in neurological research. And that is because we don't know the target. We don't know the basic pathophysiology except for some diseases like Parkinson, Alzheimer's, epilepsy, we know a little bit, but rest of the like multiple sclerosis, amyotrophic lateral sclerosis, muscular dystrophy, there are lots of neurological diseases we don't know about exact pathology. So that is the reason why there is no the molecular targets are available, so there is no innovation, nothing is happening at that level. Then definitely in case of psychiatric diseases, the diagnosis with the help of DSM-4, 5 criteria is again challenges because that criteria is not follow your underlying basic pathology of your disease. So there is a mismatch. And then insufficient sharing of data, knowledge and expertise between the academic and industry researchers is one of the things. And there is some Health Insurance Portability and Accountability Act (HIPAA) that restricts the use of the health information, disclosed information, so there is a need to do something on this. Then there is a low yield in the biomarker discovery, that is one of the barriers you can see because few validated biomarkers exist and most of the biomarkers there is no predictive value. Means biomarkers are those you can say the characteristic which have a correlation with your ultimate hard endpoints and there should be a correlation, but none of these very few biomarkers in this CNS are predictive validity. So that's why there is nothing happening in this biomarker discovery in neurology. Then high failure rates in clinical trials. So if you see, if all know by this time, thousands of compounds, they screen 10 compounds enter into the clinical phases of different phases of clinical trial, and ultimately one compound gets approval from the individual organization. So the chances is very less, and the developmental process typically takes 10 to 15 years. So these are some of the statistical data taken from one of the publications. Screening failure rate is very high in neurological research, 57% as compared to 36% in other therapeutic areas. Then mid to late stage failure is also very high in CNS at 85% as compared to other, that is 31%, and approval rate. So it is only 36% in the drugs which are studied in the neurology research as compared to 71% of oncology. So the chances of getting success in neurological research is very, very low. new compounds. So that is the reason why there is no innovation is coming in neurological research. Then high cost, I'm not going to, and unimportant challenges with neurological research is lack of clarity in regulatory requirement, because other like drug approval process very clear in the guidelines, USFDA. These are the things you have to establish. But in case of neurology research, disease like there is nothing is clear from the regulatory aspect. So what the sponsor actually did, they try to collect as much as data they want. Like some of the data are not required. So there is something mismatch. So that things to be regulatory authority should be very much into to give the guidance for the sponsor, what are the data they need to for the approval. So that area is a great with the neurological drug approval. Then advancement, just basically what are advancement with respect to the diagnosis, the prognosis, like you have the this neuroimaging techniques like functional MRI. So what this functional MRI says, actually they can measure the brain activity by changing the blood flow. So these are the representative of the MRI picture. And also active brain regions, they consume more oxygen. So they capture while patient is consuming something, what is the activity area of the brain, where there is more blood circulation, where there is more oxygen consumption. So these are one of the advancement you can say neuroradiological imaging techniques. Then PET scan you all know by using the radioactive tracer to visualize metabolic processes. So this is a scan, amyloid scan for the amyloid Alzheimer's disease patients. So positive means these areas, green and yellow area is the amyloid beta position. So negative matlab less, positive means more. So this type of things help us to say what happened to the patients with the treatment at the level of diagnosis. Then there is electron microscope. You all know about the electron microscope routine 1. So electron microscope try to capture very small cellular structure. But what is the advancement? It is a 3D advancement. Electron microscope that allow us to reconstruction of the complex neural structure. So this one is the one of the taken from publication, how this 3D electron microscope images is actually shows different color, different circuit, neuronal circuit. So it's a very advanced, you can say. Then there is connectomics. This is new one, little bit new, not very new. So what is connectome? Connectomics is a field which aims to comprehensively map the brain's connection, both the structural and the functional connection, and that help to understand us how the different regions of the brain work, physically also and chemically also functional. So this is one I have taken from one of the publications. So connectomics actually what are the create a scaffold that supports stem cell survival and differentiation. And they have also role in nanomaterials in diagnostic and imaging tools. And these are some of the nanoparticles which are targeted. And these are just some of the diseases where nanotherapeutics are used or under study, like Alzheimer's, Parkinson's, brain tumor, and stroke. Then gene therapy. So it can develop restoring the neurotransmitter synthesis in case of Parkinson, then promoting neuronal repair with the help of BDNF and GDNF. Then you can modify the neural circuit by rebalancing dysfunctional circuit, and then RNA therapies by modifying the gene expression with the help of this antisense oligonucleotide RNA interference or RNA editing. And then these are the examples where gene therapy are undergoing research, like spinal muscular dystrophy, Parkinson, Huntington, Dravet, and all this. Then cell therapy, like stem cell therapy, all know, neural stem cell therapy or mesenchymal stem cell therapy, or encapsulated cell delivery, where cell is encapsulated with your some material membranes and then implanted into the brain, and then the cell will release that particular compound or your therapeutic interest compound, and then it will give like localized release, continuous release. And then carrier cell can be a type of delivery vehicle. Cell can be modified in a delivery, and you can, and then that cell can entrap that particular molecule and then release it directly into the brain. And these are examples of cell-based therapy in serious disease: Parkinson, Alzheimer's, multiple sclerosis, stroke, traumatic brain, spinal cord injury. So these are the area where cell therapies are there. And modeling and imaging, like with the help of this imaging system, you can do the computational modeling, and you can get idea about the disease process, and you can prognosis of the that particular disease, and also the hypothesis testing of this particular disease. And lastly, the role of AI. You all know there are lots of lectures in this conference, AI. So AI has a different role in AI also. So at different level, at the diagnostic level, at the you all know, I will not discuss, but different level nowadays, AI, what they personalize the plan, create a tailored treatment plan for a particular patient based on their neurological profile. Even they can help, AI can help in surgical planning, creating a 3D model of brain, and then predict the tumor, and then remote monitoring with the help of AI powered wearable device. The physician can monitor what happens to the patient with respect to the mobility, with respect to the speech, so that all are the advancements and that discovery and details. And these are the, like, even there is a challenges, but with the help of the advancement, the future of the medical research is, I think, it is promising. So challenges is there, but promise is also there because of AI, because of latest advancement in various neurological techniques, and so on. Yes. Okay, any question? Anything? This is a very generalized topic. Actually, I am not focused because time is also not. So disease specific challenges is also different. So this is the overall challenges in neurological challenges in both in the patient and in the animal study also.
Thank you, Dr. Saha, for your comprehensive and insightful presentation. Your expertise has brought clarity to the evolving landscape of neuropharmacology and highlighted the critical areas that merit further exploration. Now I will request our chair, Dr. Aswar, to grace the dais and felicitate our speakers. Dr. Milind Parle. Dr. Vivek Jain. Dr. Lekha Saha. Now I'll request Dr. Manish Jain to please felicitate our chair, Dr. Manoj Aswal. We extend our deepest gratitude to our respected chair and esteemed speaker for their erudite contributions, and to the audience for their dignified presence and thoughtful participation. May the insights shared today continue to inspire rigorous inquiry and scientific excellence.
Good afternoon, everyone. Thank you for presenting. It is my absolute honor and a great privilege to commence with the next segment of the program, that is the open oral presentations. To chair this session, may I please welcome on stage Dr. Jai Malik and Dr. Manish Jain, sir. Please, sir, join us over the stage. It is a pleasure to introduce Dr. Jai Malik. Sir is an associate professor of pharmacognosy at University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. He holds a PhD and master's degree in pharmacognosy from UIPS, and sir has a wonderful academic and research experience. His research focuses on bioactivity guided isolation, herbal drug standardization, analytical method development, and plant-based therapeutics for CNS disorders. Dr. Malik has guided several postgraduate and doctoral scholars. Sir has secured great positions in competitive research grants and published impactful work in reputed journals. He has also received national and international recognitions, including Best Paper Awards and International Training Fellowships. His continued contributions reflect excellence in research, teaching, and advancement in the field of pharmacognosy. Thank you, sir. And now I will introduce our next chair, Dr. Manish Jain. It is my pleasure to introduce him. Sir is a Ramalingam Swamy Fellow at UIPS Punjab University. Dr. Jain is a accomplished researcher in cardiometabolic vascular diseases with international experience in Switzerland and USA laboratories. He has more than 50 publications and is honored with prestigious awards, including the AHA Young Investigator Award and the Ramalingam Swamy Fellowship. We warmly welcome both of you to chair this session. Sir, I request you to please join us in front of the stage so that we can commence with the session. So the first presenter for the day is Dr. Villiers Masoka. Sir, the stage is all yours. Please come.
Thank you. Okay, thank you very much, and I'm happy to be here, and also I'm grateful for the privilege to give a talk at this conference. I've enjoyed the conference a lot. My research is on neuropathic pain. I work at Kuwait University, and I'm in the Department of Pharmacology and Therapeutics. I'm working on a number of molecules, mostly to prevent or to attenuate or ameliorate neuropathic pain. The one I'm going to present today is lycophyllin, which is a molecule a number of people at this university have worked on in previous years. I know Professor Kukane worked on it a lot. And this drug went as far as the phase 3 clinical trials for osteoarthritis, but unfortunately the results were mixed, so it stopped at that level. But a number of people have now started looking at it for other purposes outside osteoarthritis. So the type of pain I work on is neuropathic pain, which is pain caused by a lesion or a disease of the what? Of the neurons, basically of the nervous system. There are a lot of types of neuropathic pain. We can divide them into peripheral or central. In terms of the central, we have, for example, spinal cord injury, multiple sclerosis. In terms of the peripheral, we have post-herpetic neuralgia, painful diabetic neuropathy, and the one we work on, which is chemotherapy-induced neuropathic pain. So this is the pain caused by drugs such as paclitaxel, vincristine, oxaliplatin, when they are given to patients to treat cancer, various types of cancer. In our lab, we use animal models of neuropathic pain. We use mostly rats and mice, and we work mostly on paclitaxel, but we also work on vincristine, bortezomib, and oxaliplatin. So we work on a various number of drugs. But for this conference, I'll be presenting the data from paclitaxel. And paclitaxel basically can cause mechanical allodynia. So when I say mechanical allodynia is increased sensitivity to something which is not painful. So let's say someone touches your hand and you feel pain, that is allodynia, because under normal circumstances, you are not supposed to feel pain. When we talk about allodynia, is something which is not supposed to cause pain, causes pain. I will also talk about hyperalgesia, which is basically increased sensitivity to painful stimuli. So you feel more pain than under normal circumstances, although the stimuli already causes pain. So we use, in terms of mechanical allodynia, we use an equipment called a dynamic aesthesiometer, which is basically an electronic von Frey. So it's much easier than the normal von Frey hairs, and also it reduces the bias, because all the experiment has to do is to press the button and then the filament goes up, and when the animal removes the paw, the filament stops and the time is recorded automatically. So it reduces the bias. That is one of the idea of using the electronic Van Frey. And we have shown in previous studies that animals, for example, treated with paclitaxel, when we measure mechanical allodynia, they develop mechanical allodynia from day 7 and start recovering from day 18. So there is a time period where they have mechanical allodynia. The drug which we are interested in which I'm going to present for this conference, is lycophyllin. It's a dual LOX and COX inhibitor, and it inhibits both COX and LOX almost equally. So we have done some studies with lycophyllin. I was told to have 8 slides. That's why my slides are kind of packed. So we have done some studies before with lycophyllin. So we treated animals with paclitaxel on this slide, and the animals developed mechanical allodynia. Once the animals had developed mechanical allodynia, we gave them lycophyllin at different doses, and then we measured their response. And we could see that lycophyllin basically increases the withdrawal threshold in a dose-dependent manner. And we also tested whether the mechanism, so we know it's a dual COX, LOX, but dual COX, LOX, COX also can have other effects. We are interested in the endocannabinoid system. So the endocannabinoid system is composed of cannabinoids, receptors, the endocannabinoids like 2-AG, anandamide, and the enzymes that degrade them. And there are many enzymes which degrade endocannabinoids, mostly fatty acid hydroxylase and monoamine glycolipase. These are the principal enzymes, but also COX and LOX, they do oxidize these endocannabinoids and terminate their effects. They are not the main enzymes, but they also have an effect on these endocannabinoids. So we tested whether lycophyllin had an effect via cannabinoid receptors. So we tested both cannabinoid CB1 receptor and CB2 receptors. And the data shows that the effects of lycophyllin were dependent on both CB1 and CB2 receptors, which is something which we found very interesting. And then we did molecular docking to see whether lycophyllin has affinity to these receptors. And in summary, we found that lycophyllin could dock to both CB1 and CB2 receptors, and the affinity was similar, although a bit less than THC, which is the phytocannabinoid. So we could say, lycophyllin might act through cannabinoid receptors on top of acting through inhibition of COX and LOX. And we have also done previous studies where we measured the amount of endocannabinoids in the pore skin, so in the periphery, because chemotherapy-induced neuropathic pain is a peripheral type of neuropathic pain. We measured using LCMS the amount of 2-AG, and we could find that there were no differences in the brain, there were no differences in the spinal cord, but there were differences in the pore skin. So endocannabinoids were reduced, which could be one of the mechanisms which contributes to the development of what? Mechanical allodynia. So one of the hypotheses we are looking at now is, does lycophyllin have an effect on 2-AG levels? We know that they go down. Can lycophyllin increase them through basically inhibiting COX and LOX? We know it's not the main enzyme, but maybe it could also increase the amount of what? 2-AG. So this is our experimental paradigm. So we use sprague-Dawley rats, and we treat them with paclitaxel and lycophyllin. So in this study now, which is following, it's a preventative. What I presented before was more therapeutic. So the animals have developed allodynia. Can we treat the allodynia? Now is, can we prevent? And it makes sense for a disease like chemotherapy-induced neuropathic pain, because we know we initiate it, even in human beings. We want to treat cancer, and about 70% of the patients will end up with chemotherapy-induced neuropathic pain. So it can make sense also to look at the possibility of preventing it, because you give a drug, paclitaxel, you know that 70%, 30%, 40% would develop chemotherapy. So what about if you give them another drug so that they don't develop? That is the rationale behind. So we give the drug for 5 days together with the paclitaxel. So we basically give the drug for 5 days, but we give paclitaxel on 2 days, 2 alternate days. And then we measure mechanical sensitivity before we administer the drugs and 9 days after we administer the drugs. And we measure allodynia using the dynamic aesthesiometer. Then we also measure hyperalgesia now using what we call the pressure application measurement, which is almost like a forceps where you can press on the animal and measure the force. So we have a device which can measure the force you are applying to the animal, basically. So this is hyperalgesia because we know this is a painful stimuli. And then on the 9th day, we again measure the sensitivity. We also measure the amount of 2-AG, which is in the pore skin. So we take the pore skin, we homogenize, and we measure the amount of 2-AG using an ELISA, a competitive ELISA now in this case. So in other studies, we're using LCMS, which is much more sensitive, but also expensive. In this case now, we are using ELISA, which is less sensitive, but also affordable. And we actually get this ELISA kit from India. It's an Indian company which sends this kit for us, which synthesizes this kit for us. So I presented both the allodynia and the hyperalgesia in the same slide. So the first, we are showing allodynia. So this is before we treat the animals. We can see that their response to mechanical stimuli is similar. When we, after 9 days, the ones which got paclitaxel alone, we can see that they have reduced withdrawal threshold, which means they have allodynia. The ones which were treated with paclitaxel and lycophyllin, they are almost equal to the vehicle-only treated animal. So we can safely say lycophyllin prevented the development of mechanical allodynia. Hyperalgesia, we had not characterized it before, so we characterized it in this project. And we could also see that the animals who developed hyperalgesia, and they recovered by almost day 35. Okay, I think I'm just about to wrap up. Okay. And when we also looked at the treatment with paclitaxel and lycophyllin, it was kind of similar to the what? To the allodynia. So lycophyllin prevented the development of what? Of paclitaxel-induced hyperalgesia, but it was less efficacious compared to the allodynia. And we measured the amount of 2 Ag in the pore skin, and this is the normal level in the vehicle, and we could see that there was reduction in the paclitaxel-treated animals, and there was a reversal basically in the animals which were treated with paclitaxel and lycophyllin. Of course, there was a lot of variation, which is expected because COX and LOX are not the main enzymes involved in the metabolism of 2-AG. So in conclusion, co-treatment with lycophyllin and paclitaxel prevents the development of mechanical allodynia and hyperalgesia, possibly through preventing the reduction in 2-AG. So I think it's in part, it might be also in addition to COX and LOX inhibition. And these findings suggest that COX and LOX inhibitors could be useful for the treatment of developed neuropathic pain or for prevention of neuropathic pain. In terms of acknowledgements, I would like those who worked specifically in this project. There's Amar Thomas on the far right. She's from Kerala. And then we have Esra Ali from Egypt. And then Aisha Al-Balush and Altaf Al-Romain from Kuwait. In terms of LCMS, we also have some of our collaborators in Ireland. Thank you very much. Any questions? It has worked on other types of allodynia. So, for example, I think Professor Kukan worked on incisional allodynia and some of nerve ligature allodynia, and they've shown that it might be useful as well.
And what about the consequences of inhibiting these COX logs? So, I just want to know about this prostaglandins. Some of these prostaglandins, they are beneficial also, and if they inhibit these COX logs both, so prostaglandin production will also be inhibited.
Okay, actually, the interesting part is inhibiting both COX and LOX much better than COX alone.
Yeah, that understand, because otherwise it will be shifted towards LOX pathway.
Yes, and even it was shown to be gastroprotective compared to indomethacin. So if you give, for example, indomethacin or ibuprofen in an experiment, they will develop ulcers. If you give lycophyllin, they don't.
In addition, they will predispose a person towards asthma, because most of this COX pathway will be shifted towards LOX pathway.
Yes.
So I was just asking this, if we inhibit both COX, LOX, so all the prostaglandin production is ultimately shut down. So is there any consequences of those preventing prostaglandin?
So far,
what's their production in platelets?
I mean, there are a number of things which could happen, which could happen with all course they have side effects, but they are being compared with drugs already in the market.
I have also one question, because most of the anti-cancer drugs, they basically cause a drastic alteration in the liver function biomarkers.
Yes.
Isn't it?
Yes.
And we are supplementing it with anti-inflammatory drug, which is again going to affect the liver biomarkers.
Yes.
So how are you going to tackle that situation?
So the idea is, if you, because normally you give them for a short period,
okay,
so you are definitely going to alter the liver function.
But paclitaxel is not going to be given only in 2 doses, isn't it?
Yeah,
While treating the
it's,
cancer.
it's a number of cycles.
Right.
Exactly. But it depends. Our actual proposal, we are working on something which we have started, is to give them peripheral. So to develop a dosage where you give them, because normally paclitaxel causes neuropathic pain in a glove and socks manner. So if you could develop, let's say, creams or gels, because now there are some drugs like lidocaine and the capsaicin which are given for these patients topically.
That is basically anesthetic, local anesthetic.
Yeah, but capsaicin as well is given.
Capsaicin has a different mechanism.
Yes, so I'm saying we can also try to use the same protocol where you inhibit the endocannabinoids specifically, peripherally, you reduce the toxicity issues.
But again, to target whole of the body, or to put cream on whole of the body.
Sorry?
To put cream whole of for all whole of
No, most of the patients, their major problem with the chemotherapy is, yes, very localized. I mean, they will have other pains like any cancer patient, but when we took a neuropathy, most of it is purely glove and socks. Even when you go to books, it's described as gloves and socks. And even when you go to cancer hospital, because sometimes we visit cancer hospital, most of the patients, their major issue is the burning sensation, the electrical sensation, the pins, and it's on the feet and the hands. So you are trying not, you are not going to prevent, but reduce. Most patients actually stop taking paclitaxel because of chemotherapy, or they reduce the dose. So if you could give something which makes sure that the patients can go through the whole course of cancer of chemotherapy, I think that would be a big advantage.
Hello. My topic for today is evaluation of prescription patterns and self-medication practices in patients of osteoporosis. It was a cross-sectional questionnaire-based study. Now, prevalence of osteoporosis in the world is 18.3%, and in India it is 22.9%. Why I wanted to study prescription pattern is because treatment of osteoporosis involves many drugs, which leads to drug variability. And as osteoporosis is a chronic disease, a lot of patients resort to self-medication practices, and hence I wanted to evaluate it. My primary objective was to evaluate prescription patterns of drugs used in treatment of osteoporosis, and to assess self-medication practices in patients of osteoporosis using a pre-validated self-medication questionnaire. It is a cross-sectional questionnaire-based study. We obtained IEC approval and registered on CTRI. We followed duration-based sampling for 11 months. We took participants from endocrine and orthopedic OPD of a tertiary care hospital. Then according to our selection criteria, after seeing it, then we took written informed consent of the patient. Demographic details of the participant were noted, and the prescription details were noted in the CRF, and then a pre-validated self-medication questionnaire was asked to the patient. So our inclusion criteria included male or female, age more than 18 years, attending endocrine OPD or orthopedic OPD, and they have been receiving treatment for at least 1 month, and who have a confirmed diagnosis of osteoporosis. Exclusion criteria were patients who are not willing to give informed consent, patients in IPD and emergency, and newly diagnosed and treatment naive patients. So the variables that I measured, primary variables were WHO prescribing indicators, percentage of incomplete prescriptions, the most common drug class, the most common drug in each class, the rational and irrational FDCs, PDD-DDD ratio. And my secondary variable was the percentage of participants who said yes for the self-medication questionnaire. Now coming to statistical analysis, continuous variables would be represented as mean and standard deviation, categorical as percentage, variables of prescription pattern and self-medication questionnaire as descriptive stats. Now, we recruited 250 participants with mean age of 60.46 plus minus 9.650. Majority were females, that is 93.6%. Total number of prescriptions were also 250. The total number of drugs prescribed were 435. So average number of medications for osteoporosis per patient was about 1.63 plus minus 0.55. So a majority of the drugs, that is 70.80%, were prescribed by their generic name. And 98.60 were prescribed from WHO essential drug list. Now coming to the prescription analysis, the percentage of incomplete prescriptions were 23.6%, out of which dose was the most common missing factor. The percentage of FDCs was 7.6, that is 19 out of 250. All were rational FDCs. Calcium was the most common prescribed drug, that is 98.4%, and vitamin D 70%. The PDD-DDD ratio for vitamin D was 2.5, that was slightly overprescribed, and for zoledronate it was 0.8. Coming to my results for self-medication practice questionnaire, 6.8% of patients showed that they were resorting to self-medication practices. They said they were usually using it as an add-on. Out of that, out of 17, 12 were resorting to Ayurvedic and 5 for homeopathy. They would take it once a day. 14 would apply topical and 3 would resort to oral. The reason for taking it was stronger bone and pain. About 3 were only aware of the side effects, 14 were not aware, and they learnt about these medications from family, friends, or social media. And 10 out of them felt that it was necessary to inform their doctor. Coming to my conclusion, almost all patients were prescribed calcium and vitamin D, and the next common medication prescribed was bisphosphonate, zoledronate. Nearly 1/4 of the total prescriptions were incomplete, and most common incompleteness was in terms of dose, and only a small number of patients currently resort to self-medication practices for osteoporosis. These are my references. Thank you. Yes, sir. 9 questions. Yes, sir. We had 3 validators from each department. So we calculate the CVI ratio, and that's how we validated the questionnaire.
Was there any validated questionnaire available?
No, sir. I couldn't find for self-medication practices.
So according to your data, calcium is being taken?
Most common. No, it is with prescription. Dose is missing. Thank you.
Thank you, ma'am, for the presentation. So the next presenter would be Dr. Sushil. Please come on stage.
Good afternoon, everyone. My research topic is study of determinants of quality of life in patients with ischemic heart disease. So ischemic heart disease is one of the leading causes of premature mortality and disability in developing countries like India. The most common clinical manifestations are chronic stable angina, STEMI, NSTEMI, and unstable angina. Quality of life, a predictor of general well-being, is an independent risk factor for mortality and major cardiac events. Multifactorial factors like patient, disease, and therapy influence the quality of life in patients. Hence, identification of these factors help in developing of a counseling tool to improve health outcomes. Aims. The aim of the study was to study the determinants of quality of life in patients with ischemic heart disease.
Objectives was to assess the quality of life, then find the association of defined risk factors and quality of life, and to develop a counseling tool for improving quality of life based on risk factors in patients with ischemic heart disease. Material and method, the study was conducted after approval from the ethics committee. It was a prospective type of observational study done in Department of Cardiology and Pharmacology of MGM Medical College, Navi Mumbai. Total sample size of 250 patients. Inclusion criteria had patients of either gender, more than 18 years of age to 75 years of age. Known IHD patient attending cardiology OPD for more than equal to 3 months, patient with chronic stable angina or acute coronary syndrome, and patient willing to participate. Any other structural heart disease patients were excluded. Evaluation parameters, quality of life among patients with IHD were evaluated using health-related quality of life questionnaire, and then they were categorized into 2 subgroups of better quality of life and worse quality of life. Now, determinants of quality of life had disease-related factors like diagnosis and comorbidities. Therapy-related factors had type of medication and medication adherence. Patient-related factors had socio-demographic profile, anthropometric and metabolic profile, lifestyle, sleep quality and sleep time, screen time, self-care monitoring. And then third, development of a counseling tool by association of various risk factors, and quality of life was determined by comparing between high and low quality of life groups, and a counseling tool was developed. Now results. Among the total 250 patients, 60 had better quality of life, that is 24%, and 190 patients had worse quality of life, that is 76%. Now, patient-related factors affecting quality of life, gender, males were more prone to ischemic heart disease, but females were more prone to the worst quality of life. Body mass index, obese patients were more prone to have the worst quality of life. Sleep duration, people with worse health-related quality of life had less than the recommended sleep hours of 7 hours. Sleep quality, patient from a worse quality of life group had poorer sleep quality as compared to patients from better quality of life. Then various other factors like age, socioeconomic class, abdominal fat, self-care monitoring, etc. were the non-significant factors. Then in lifestyle and screen time, average fat intake, average salt intake, alcohol intake, and average screen time were the significant factors. In disease-related factors, clinical diagnosis, the most common clinical diagnosis we found was STEMI. Comorbidities, people with dyslipidemia were more prone to have the worst quality of life among the patients of ischemic heart disease. Patients with better quality of life were found to be more adherent to their medications. Then beta blockers and biguanides were the most common drug that were prescribed among the patients. In better quality of life, we found HMG-CoA reductase inhibitors like atorvastatin to be the next most commonly used drug. While in worse quality of life people, we had potassium channel openers. This is the, we have developed a counseling tool for improving the quality of life. It explains the quality of life and why do we need to assess it. According to the factors like gender, lifestyle modifications, dietary habits, addictions, and medication adherence. Gender, like BMI has to be maintained between 18.5 to 23 kg per meter square by having high fiber diet and avoiding processed food. Hypertension controlling less than 140 by 90 mm of Hg. Screen time less than 2 hours and to be Conclusion, for Objective 1, assessment of quality of life, 76% of the patients of ischemic heart disease reported worse quality of life. Objective 2, association of the risk factors and quality of life. In patient-related factors, significant association was found with gender, BMI, sleep duration, and sleep quality. Disease-related factors, diagnosis, and presence of comorbidities were significant determinants of quality of life. Therapy-related factors, medication adherence had a positive association with quality of life. Objective 3, that is development of the counseling tool, it was successfully developed to improve the quality of life and will later translated into various regional languages to improve the patient education and adherence. We acknowledge the funding from ICMR for the PG thesis 2023. Thank you. So we used the Kupuswamy scale, but it didn't come significant while statistical analysis. So. Yes, ma'am. Sir, actually, it should be. Sir, actually, the patients with worse quality of life are of significant number. Out of 250, there were 190 patients. So that's why the mean, both the mean came to be very close. That's why. Otherwise, the yeah, yes, yes, sir. Yes, yes, sir. Yes, sir. Thank you, sir.
No folder, just clean. Yes.
In the wild world of pharmacotherapeutics, finding a new drug is like finding a rare star. It often takes billions of dollars and a decade of time to find a new therapy. Traditional drug discovery is like a marathon of uncertainty. If we screen 10,000 molecules, hardly one comes into therapeutic practice. What if the cure that we are looking for already lies within the drug shelf, waiting to be reimagined? Human body speaks the language of genes. Some genes are upregulated or downregulated in a disease. This is known as genetic signature of the disease. What if a molecule can reverse this genetic signature? Would it be a potential therapeutic match? Good morning, everyone. Respected doctors, seniors, dear colleagues. Myself, Dr. Abhiram, third year pharmacology resident, Department of Pharmacology, Lady Hardinge Medical College. The topic of my presentation is AI-driven drug repurposing, genomic signature matching for evaluation of potential neuroprotective drugs in Alzheimer's disease. Today we are going to explore the following subjects: introduction, aims and objectives, methodology, results, discussion, and future perspectives. Alzheimer's disease is a chronic, progressive, neurodegenerative disease affecting more than 55 million people worldwide. It is characterized by cognitive decline, behavioral changes, and memory loss. It is pathologically characterized by tau neurofibrillary tangle deposit, beta amyloid plaque deposit, and a component of neuroinflammation. The current therapeutics include cholinesterase inhibitors, mementamine, anti-amyloid monoclonal antibodies, etc. But the current therapeutics is often inadequate in preventing the progression of the disease. So the search for the new therapeutics is always going on. But what if I tell you, if we can screen the already approved drugs in a serial manner to look for neuroprotection and the ability to reverse its genetic signature? Today we are going to exactly discuss the same. This is known as genomic signature matching. So genomic signature matching includes, there is a disease signature. As I said earlier, every disease has, in a disease condition, some genes are upregulated and some genes are downregulated. This is known as the disease signature. And when we introduce a drug into the body or a cell line, it also causes upregulation and downregulation of certain genes. This is known as the drug signature. So what we are trying to do is here that if the drug signature is able to reverse the genetic disease signature, it would be a potential therapeutic starting point for drug repurposing. So we have the following databases. The first database is that the LINC L1000. This is maintained by the MIT and the Harvard. It contains the genetic data, that is the transcriptomic data of the drug. When a drug is introduced into the cell line, the upregulated genes and the downregulated genes, this data is provided by the L1000 level 5 evidence. So what this database does is that they do microarray experiments in the cell line. So they do 938 gene ka expression. Out of this 938 genes, they extrapolate it to 12,000 plus gene using machine learning algorithm. And now for the disease database, we have something known as the Gene Expression Omnibus, which is a repository of the gene expression data from the studies around the world. This is maintained by the same database which maintains the PubMed, that is National Center of Biotechnology and Information. My primary objective is to identify the transcriptional signatures of the disease, followed by the evaluation of the drug candidates and their transcriptional signatures, followed by signature matching and to perform the connectivity analysis, and followed by to identify the neuroprotective drugs. My secondary objective includes identification of the top up and down regulated genes, conducting something known as the FSGEA pathway analysis, which means that fast gene enrichment analysis, and to do a differential expression of the gene, followed by the visualize the connectivity trends. My methodology starts with finding the data, finding the GEO, Gene Expression Omnibus datasets. I did it through a package in R known as GEO query. And I got the two datasets, that is GSC48350, GSC5281, which contains the postmortem samples of people with Alzheimer's disease. So what I did next is that I converted the probe ID of the samples into the genetic ID using annotation DBI, which is an extension in R software. And I integrated the two Alzheimer's disease databases via meta-analysis and I combined the set scores to get the Alzheimer's disease signature. The next step was to obtain the drug signature, which I got it from the LINC 1000 level 5 database. It is level 5 because this is a processed information. I did it with the help of RHFD package in R, followed by the Lima package to normalization and empirical base correction to get the differentially expressed genes and to find the log-fold2 change and the false discovery rate, followed by doing the connectivity analysis and conducting the pathway enrichment with FSGEA and to visualize the results. The key drugs investigated in this study are vorinostat, which is an histone deacetylase inhibitor, resveratrol, which is an antioxidant and sirtuin 1 activator, sirolimus, which is an mTOR inhibitor, metformin, which is an AMPK activator, and transpimycin, which is an heat shock protein 19 inhibitor. Coming to the results, first I performed the connectivity analysis. In the connectivity analysis, we get something known as the Z score. The more negative the value of the Z score, the more potential the drug has to reverse the genetic signature of the disease. So here we can see that after performing the connectivity scores, here we can see that vorinostat and resveratrol has the highest negative median connectivity scores, indicating a potential disease reversal by the drug. While we can see that metformin and sirolimus has a positive connectivity score, which indicates disease mimicking trends. And transpimycin has a mild negative connectivity score indicating partial reversal. Coming to the next pathway enrichment using FSGEA, we get something known as NES, that is the normalized enrichment score. If the normalized enrichment score is positive, the pathway is positively enriched. And if the NES is negative, it will be negative enriched. So after getting this biological pathway, we found out that the positively enriched pathway in Alzheimer's disease include fatty acid metabolism, complement, etc. And the top negatively enriched pathway include peroxisome, unfolded protein response, etc. Now what we did is a similar pathway enrichment work conducted for the vorinostat and resveratrol. As we can see here that the positively enriched pathway in the Alzheimer's is reversed by vorinostat and resveratrol with varying degrees. Now coming to the volcano plot analysis, here we can see that the x-axis contains log two-fold changes. The y-axis contains the Z score. Okay? So log fold 2 changes means that how many times the genes are upregulated or downregulated. If it is positive, it will be upregulated. It will be negative, it is downregulated. And the Z score says that the negative the Z score, the more potential it has to reverse the genetic signature of the disease. So we can see that on the left lower axis, we have the downregulated genes which can reverse the disease signature. And to the right lower part, we have the upregulated genes which can reverse the disease signature. Coming to the discussion, the top candidates we got is that vorinostat and resveratrol has a most negative score, followed by sirolimus, which has partial reversal. And the positive connectivity scores were received by metformin and transpimycin, which indicate disease mimicking trends. And we can see that it is not only the genetic upregulation and downregulation, but also the pathways. The biological pathways are also affected. The prominent pathways which is reversed are MYC target, hypoxia signaling, etc. My limitations are, these are non-neural cell lines, because LINC database uses cancer cell lines. It is heterogeneous data because postmortem samples were taken from different parts of the brain. And we know that Alzheimer's disease is a chronic disease, but LINC database looks the gene expression after 6 to 24 hours. And transcriptomic reversal doesn't always mean clinical efficacy because it doesn't have PKPD data or blood-brain barrier penetration data. And lack of post-translational data like proteomic and metabolomic data is missing, and the lack of mechanical validation. And the future perspectives include, we have to use a neuronal models like the induced pluripotent-derived stem cells. If we integrate multi-omics database like proteomics, it will be better. And to make a network-based machine learning models, and to do an in vivo validation using pre-senilin models, etc. And finally, to do a clinical validation. I would like to conclude by saying that by decoding the language of the dysregulated genes, transcriptomic drug repurposing allows us to pair existing medicine with new diseases, guided not by chance, but by molecular biology. Thank you, everyone. Yes, sir.
Prevalence is significantly higher in patients at the age above 80 years.
Yes, sir.
Started from 16.
Yes, sir.
60 years and above, but prevalence significantly increases when the patient crosses 80 years of age.
Yes, sir.
And most of the diagnosis is done at that stage of the age.
Yes, sir.
Isn't it? So if we are giving the gene modifying therapy, though you have used resveratrol has been amply studied by the scientific community for its effect on gene regulation, specifically in aging.
Yes, sir.
So I don't think that is a repurposing of resveratrol.
Yes, sir.
What is used as an nutraceutical and phytopharmaceutical.
Yes, sir.
It has significant studies on ADI. Now, as we were discussing that the diagnosis largely is done
Yes, sir.
So if we are going to give gene modifying therapy at that stage of life, do you think that would be successful, or it will just mitigate the symptoms or provide you a symptomatic treatment?
Sir, you're exactly correct, sir. We are actually diagnosing at a late stage or after the symptoms approval. So if you use a gene modifying therapy, like, so the pathological changes has been already been occurred. And and the neuronal, it says that the neuronal cells don't divide, but there is something known as neuroplasticity, but it is shown in only typical parts of the brain. So I think those therapies would change the dividing cells, but not the pathology which has already occurred.
So can it be used if we go for gene mapping or genetic studies in the early stages of life?
Sir, Sir, that is, we are still at a dilemma of that. We can do, there are many single nucleotide polymorphism which can contribute to the development of Alzheimer's disease. So nowadays, before, I will just start this with an example. Before the AI revolution, we developed machine language models. We were developing small targets so that we will ultimately build something bigger. So now we are mapping, with the genetics, we are mapping single nucleotide polymorphism from starting. And if we could get a bigger picture, why this is happening with all this polymorphism, maybe at an earlier point, we can start a therapy which will target multiple pathways or multiple receptors which could mitigate this disease in the later stages. Yes, sir. Yes, sir.
So if you have a familial history of such kind of genes that are upregulated during the disease, so there is every possible chance the present generation might develop this disease at later stage. So can we start therapy much earlier?
As you said, it is a good possibility. Like I also thought that if these genes are upregulated, if we could start, if we have a familial history, if we could start this therapy earlier, these genes will stay suppressed so that the disease will not develop. But I'm not completely sure that we still don't have the enough proof to do so. But this is like a very good idea to think about in the future researchers. Longer time.
Thank you so much to all the presenters. Now I request the chair of the session, Dr. Manish, and Dr. Jamalik, sir, to please join us on the stage to present certificates for the presenters of the day. Please, sir. Dr. Villiers, please. Dr. Urjita Kapadia. Dr. Sukshiti. Dr. Abhiram Suresh Babu. Now, I would request Ms. Mini and Dr. Jatinder Dhaliwal from our organizing committee to please felicitate the chair of the day, Dr. Jay Malik. I will also come in this. Dr. Manish Jain. Sir, a collective photo with both of you. So, okay. So can we have a group photo with the presenters also? I request all the 4 presenters to please join us on stage. I request all the volunteers for Hall 2 to please join us on stage. Please join us on stage for the photograph, please. Ask everyone who is there outside, please. Thank you everyone for joining. Now we'll proceed to the lunch session, and after that there will be a common event in the law auditorium, 2:30 onwards. Thank you so much.